Saya juga mau ikut itu.
Sudah masih registrasi.
Ada nggak nih? Ini tuh saya ikut menarik tapi ya dibayar sertifikatnya 200.000 tidak bayar. Tidak ada pemberitahuannya itu dibayar.
Tidak ada. Perekonomian yang maju dan terpenuhinya kebutuhan pangan masyarakat seringkali menjadi tolak ukur sebuah bangsa yang sejahtera. Sektor peternakan memiliki peranan penting untuk mewujudkannya di Indonesia. Maka, dibutuhkan sumber daya manusia yang kompeten di bidang peternakan, terutama yang mampu mengembangkan teknologi untuk menghasilkan produk ternak yang berkualitas. Dalam menjawab tantangan tersebut, pada tahun 1963, Institut Pertanian Bogor mendirikan Fakultas Peternakan bersamaan dengan program studi ilmu produksi ternak. Saat ini, namanya telah berganti menjadi Teknologi Produksi Ternak. Dengan menekankan pada kemampuan menerapkan, mengembangkan, dan menciptakan ilmu di bidang produksi ternak, program studi teknologi produksi ternak ditujukan untuk memenuhi kebutuhan masyarakat. Program studi teknologi produksi ternak Fakultas Peternakan IPB University telah mendapatkan akreditasi A dari BAN PT pada tahun 2018 dan terakreditasi ASEAN University Network pada tahun 2013 sebagai bukti komitmen dalam menjaga kualitasnya. Lulusannya diharapkan memiliki kompetensi utama agar dapat bersaing di dunia global, seperti analytical thinking, entrepreneurship, technology, dan effective communication. Proses perkuliahan mahasiswa didukung oleh tenaga pendidik yang berkualitas. Saat ini, jumlah dosen program studi teknologi produksi ternak ada 37 orang, terdiri dari 7 bergelar profesor, 22 bergelar doktor, serta 8 dengan gelar master yang merupakan lulusan perguruan tinggi dari dalam maupun luar negeri. Lingkungan belajar program studi ini dilengkapi dengan berbagai fasilitas, mulai dari ruang kuliah yang nyaman, laboratorium yang modern, rumah pemotongan hewan, unit evaluasi karkas, unit analisa molekuler, hotspot area, perpustakaan, dan unit usaha komersil peternakan sebagai tempat latihan mahasiswa. Program studi teknologi produksi ternak menjalin kerjasama yang kuat dengan berbagai kampus di luar negeri seperti Taiwan, Australia, dan New Zealand. Sebagai upaya dalam meningkatkan kesempatan mengenyam pendidikan tinggi, terdapat banyak beasiswa yang ditawarkan bagi mahasiswa yang memiliki prestasi akademik atau kurang mampu dalam segi ekonomi. Nantinya, lulusan program studi teknologi produksi ternak dapat bekerja di instansi pemerintah, swasta, sebagai tenaga pendidik atau peneliti, maupun menjadi pengusaha di bidang industri produk peternakan.
Perkenalkan, nama saya Agus Hendra Gustiawan, saya alumni dari Fakultas Peternakan. Untuk saat ini saya menggeluti usaha yang saya beri nama PT Sugeng Jaya Group yang bergerak di bidang pakan hewan pada khususnya. Yang basicnya adalah serangga atau ulat Hongkong. Saya ucapkan terima kasih kepada Departemen Ilmu Produksi Teknologi Peternakan karena dari situlah kita dibesarkan, dari ilmu-ilmu yang diberikan, lingkungan yang mendukung, dosen-dosen yang humble, sehingga kita bisa menerapkan ilmu-ilmu yang sangat aplikatif. Di sisi kuliah juga kita selalu dibubuhkan semangat-semangat entrepreneur, pembicara-pembicara yang sudah sukses dan lain sebagainya, sehingga bisa membentuk mahasiswa atau alumninya ke depannya untuk menjadi wirausaha.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Gulam Halim Furqoni, saya alumni IPTP 48. Aktivitas sehari-hari saat ini saya sebagai pengusaha. Saya adalah Direktur Utama dari PT Trafinesia Muda Berkarya yang bergerak di bidang pariwisata serta teknologi. Yang saya dapatkan di ilmu produksi dan teknologi peternakan, ilmunya sangat banyak dan sangat bermanfaat. Buat teman-teman semua yang ingin berkuliah di IPB, khususnya Fakultas Peternakan, masuklah ke IPTP atau Ilmu Produksi dan Teknologi Peternakan. Sukses untuk semuanya. Wassalamualaikum warahmatullahi wabarakatuh.
Selamat datang di program studi teknologi hasil ternak IPB. Jika kamu ingin sukses di bidang pengolahan hasil ternak, maka program studi ini adalah pilihan yang tepat. Kehadiran program studi teknologi hasil ternak menjadi perluasan keilmuan sekaligus pelengkap hulu hilir Departemen Ilmu Produksi dan Teknologi Peternakan yang tidak hanya bertumpu pada produksi, namun juga ke arah teknologi hasil ternak. Mahasiswa program studi teknologi hasil ternak IPB dibekali dengan 3 ilmu utama, yaitu komoditi ternak yang terintegrasi pada proses hulu sampai hilir, penerapan teknologi untuk meningkatkan nilai tambah, dan mengoptimalkan produk hasil ternak sebagai sumber pangan hewani yang berkualitas. Program studi teknologi hasil ternak IPB telah terakreditasi B oleh BAN PT pada tahun 2018. Proses belajar mengajar dibimbing oleh para dosen yang kompeten lulusan perguruan tinggi dari dalam maupun luar negeri. Para dosen memiliki gelar doktor, master, serta ada pula yang menjabat sebagai profesor. Selain itu, juga didukung oleh 4 dosen dari luar program studi untuk memperkaya wawasan mahasiswa di bidang teknologi. Program studi teknologi hasil ternak IPB juga dilengkapi dengan berbagai macam laboratorium, mulai dari laboratorium pengolahan hasil ternak sampai laboratorium organoleptik. Meningkatkan mutu pembelajaran, program studi teknologi hasil ternak menjalin jejaring yang kuat dengan berbagai universitas di berbagai negara seperti Singapura, Jerman, maupun Jepang. Prospek lapangan kerja bagi lulusan program studi teknologi hasil ternak IPB sangat luas. Di antaranya, wirausaha pada bidang pengolahan pangan hasil ternak, technical support dan research and development pada industri peternakan, serta peneliti produk peternakan.
Perkenalkan teman-teman, nama saya Muhammad Fauzan Alfikri, alumni teknologi hasil ternak angkatan 53. Saat ini saya sedang berada di kota New York, tepatnya di Times Square, negara Amerika Serikat. Nah, pengalaman saya selama kuliah di THT, saya banyak mendapatkan pelajaran, baik itu yang berkaitan dengan bidang kuliah maupun non-bidang kuliah, seperti soft skill untuk komunikasi, negosiasi, bahkan sampai ke bisnis. Nah, bagi teman-teman yang memang tertarik di bidang pengolahan pangan, tidak hanya pangan, namun untuk skill-skill lainnya di bidang bisnis maupun di bidang-bidang bahasa ataupun bidang-bidang lainnya, bisa banget daftar di teknologi hasil ternak.
Iya, kadang-kadang tapi kakak ya. Bisa kan abang ya. Hitam. Kamu pernah makan nggak? Kamu jalan dulu be. Jalan dulu. Tahu aku tidak tahu. Tidak aku boleh balik. Aku suka aku. buat es ini ya, baru mau jual juga
Bu Herlian, maaf Ibu, Ibu share screen ya. Punten Bu, di stop aja Bu. Ibu Herliani.
plastik. Air.
Bapak Ibu mohon maaf bisa di mute dulu ya supaya suaranya yang akan muncul tidak terganggu. Terima kasih.
Halo, Assalamualaikum. Bisa dengar suara saya semua?
Waalaikumsalam warahmatullahi wabarakatuh.
Okay. All right. I think then I should switch my the language to English. I think it's just the time to start our webinar today. I saw Dr. Das is here already. Give me some few seconds. My computer, just a moment. Right. Okay. And also Dr. Aminul Islam is also here. Is it? Hello, doctor. Are you there? Okay. Dr. Nasir, are you okay?
Yes, thank you.
Okay. All right, okay. Let's begin. Bismillahirrahmanirrahim. Respected Deputy Director of International Program, Dr. Dazia Hunabi, who also representing IPB University Management and Rectorate, and also respected Professor Dr. Irma Isnavia Arif, Head of Department of the Department of Animal Productions and Technology, Faculty of Animal Science, IPB University, who also will represent the Faculty of Animal Science today. Respected speakers, we have Prof. Mehmet Ulas Sinar. I hope I correctly pronounce his name. From Turkey, he's not here yet. And then Dr. Aminul Islam from Bangladesh, and Dr. Muhammad Jasimuddin from Bangladesh, is of course our internal speaker, Dr. Ashraf Gunawan. And lastly, all beloved participants, Assalamualaikum warahmatullahi wabarakatuh, and very good morning to all of you. I would like to welcome you firstly to the first webinar, the fifth webinar series of Department of Animal Product and Technology, which will run, of course, this morning by roughly within 2-3 hours, involving 4 international speakers. Let me share the slides for you. Okay. All right, okay. I hope everyone can see my slide. All right, okay. So today webinar will took the title of Generations, Handling and Publishing Next Generation Sequencing Research Data. We consider this one as the first international webinar series of the department. For your information and also maybe for Dr. Daza information, the department has run 4 consecutive webinar series before this, but this will be the first time the series will be conducted fully in English, because I think we invite the international speakers for today's webinar, and also that's why we consider this one as international sessions. How international today's webinar actually? Okay, let me give you some trivia. We have 3 international speakers out of 4 speakers. We have Prof. Mehmet, and then the 2 speakers from Bangladesh, and of course, as usual practice in our speaker, also we highlight also the prominent staff or prominent researcher from the department. So we have Dr. Asep also, who will also share his expertise and knowledge this morning. Okay, how about participant-wise? Let's see. Out of 190 participants, surprisingly, we have participants from all continents in this planet. We have from Australia, we have from Europe, we have from African, and also we have from the States, another part of the globe. Of course, we don't have participants from the Antarctic region yet, but I think this one is enough to represent the internationality of this webinar. And gender-wise, I would love to say also that the participants are dominated by female, which is good. Yeah? I believe that this webinar will have a good support from feminist group maybe. And then the topic is quite specific, I believe, and I think if we see the distributions, the occupations background of the participants, we see that more than 80% are coming from the academic background, yeah? While the others, about 13% are from the others occupations. I think that's all for demonstrating to all of you that why we consider this webinar as international event. So I think without further ado, I would like to start this event. But before that, I would please allow me to share some housekeeping information and also regulations, as well as the detailed rundown of today's webinar. All right, okay. For the housekeeping information, some basics regulations that I hope that participants can take a note. For those who are joining, already joined the webinar before this, I think you're already aware that please use the same username for the Zoom, the same username that you use also for the registration, because basically when you log in into the Zoom, you need to be moderated, and we are trying to avoid any unknown names joining the event. And then, of course, this is from your side, ensure your internet connection is stable. Yeah, find any spots, any corner around you to get the stable connections. And then, if you look at in the chat room later, the committee will always pop up a message to request you to register yourself at the link that will be circulated in the chat room of this Zoom. Please register yourself there, particularly for those who need e-certificate after this webinar. And if you have any questions that are related to presenters, there will be a designated Q&A session. However, you can start posting your questions in the chat room with the following format: mentions your name, and then which presenter you would like to ask. Yeah, mention the name of the presenter that you would like to ask, who would like you to ask, and then your questions. Try to simplify your questions so that it's easy for us to compile and also to convey to the participants. For your information, the participant will not read your questions directly. Instead, me as moderator will compile and then convey to the speakers. And then 15 minutes before the sessions ends, all the sessions ends, I would like to seek your cooperations to activate your video for the documentation purpose. Yeah? So then the committee will get the screenshots of all of the attendance today. Okay, so I think that's all the simple regulations for this morning. And then what we'll have today is after my short introductions, the webinar we will have a short opening speech from the head of department of Animal Product and Technology by Professor Dr. Irma Isnawi Arif, who also will represent the Faculty of Animal Science, and followed by officials opening speech by the Deputy Director of International Program of IPB University, Dr. Dazil Navi. And then after that, we can start the sessions. We have 4 speakers who will be grouped into, divided into 2 sessions. I think it will be very tiring also to have a 4 talk in a row, so that we decide to make it in 2 sessions. Okay, 2 separate sessions. The first sessions will present Dr. Asep Gunawan, who will give a talk on how to perform and design RNA sequence and experiments, followed by Dr. Muhammad Aminul Islam from Bangladesh Agriculture University for bioinformatic analysis on the transcriptome data obtained from RNA-seq or microarray. And then the first sessions will be end up with the Q&A session and also the appreciation session for those speakers. And then the second session will be started with Prof. Mehmet Ulasenar from Turkey. I think he is now in the States today, who will talk about basic principle of qRT-PCR validation for holistic RNA expressions, followed by Dr. Muhammad Jasim Udin from Bangladesh Agriculture University on the trick how to publish research data and some tips on what should we do with the publications of the data. Similarly, with the first sessions, we will also close the second sessions with the Q&A and also appreciation session. And at the end, we will come up with the closings and documentation. All right, for this documentation, this is the one that I mentioned earlier. I hope that you can activate your video for 15 minutes, well, for a few minutes, I mean, within 15 minutes before the end sessions. Yeah? I think that's all for overview of the today webinar. Am I still sharing my slide? I should stop this one. Just a moment. Okay, I think, all right, okay, never mind. Okay, we have now 82 participants in the Zoom already out of 190. Maybe some of them also joining us through the YouTube channel. So we supposedly have close to 190 participants, but now we still have 82. I think many other are coming to the Zoom, so they are already in the YouTube. For your information, also today webinar are also available in the online, available in the YouTube channel. All right, okay, I think to start, I would like to invite Professor Dr. Irma to give us the speech as an opening of this webinar. Prof. Irma, the time is yours.
Thank you, Dr. Cahya. Assalamualaikum warahmatullahi wabarakatuh, and very good morning to everyone. On behalf of the Department of Animal Production and Technology, Faculty of Animal Science, IPB University, it is my honor to extend our warm welcome and sincerest gratitude to all speakers and participants of this webinar. I believe we are on the same page to state that this pandemic is quickly changing to the world landmark, not to mention research and academic activities. But it does not mean science should be stopped. The pandemic should not let our creativity in research, also academic down. By any means, the thing must keep going as these are important for our future generations, empowering them with sufficient knowledge and skills. Coming up from this background, the department initiated this webinar, whereby we translate the platform from knowledge sharing from face-to-face to the online programs. The idea to educate people and knowledge sharing. For us, important to keep proceed under any problems. And I proudly say that after several series of our webinars organized by our department, we have very good responses from all participants, which reflect that we are in the good track of having these activities, I mean webinar program. Do we have a problem? Of course, yes. Nothing comes in a perfect set. Nothing satisfies everyone. Limitation in connection, also dealing with speakers and so on. But thanks to the effort by the committee led by Dr. Asep Gunawan, those constraints were addressed accordingly. It is not only from our side, but also from the speakers and participants' cooperation. I would like to thank the speakers here who come from different parts of the globe. We have Professor Dr. Mahmud Ulasenar from RGS University, Turkey, and also now in the Washington State University, Associate Professor Dr. Muhammad Jasim from Bangladesh Agricultural University and now Queensland University, Australia, and also Associate Professor Dr. Aminul Islam from Bangladesh Agricultural University and Tohoku University, who will share their research works and experiences in producing high-impact publications from next next-generation sequencing data. My highest appreciation goes to all of you, since I believe some of you are in the different time zone, which may not be comfortable enough for you to deliver the talk. Thank you very much. But your acceptance on our schedule is a wonderful gift for us. I do believe this is also due to a good relation with our internal speaker, Dr. Asep Gunawan, who also will share his experiences in similar topic in this webinar today. This webinar is also welcome without continued support from the faculty and university management. I would like to thank the Dean of Faculty of Animal Science, IPB University, Professor Dr. Sumiati, and Dr. Dasa Hunayfi, Deputy Director International Program, IPB University, representing IPB Directorate, for always supporting us and being here with us. Thank you, Pak Dase. Lastly, to all wonderful participants, thank you so much for your support. I also noticed that some participants are called loyal participants, as always join in any series of our webinar, 1 until 4. I believe you would enjoy all talks today. Allow me to put an expectation today for this webinar, for not only as knowledge sharing, but also initiate further real collaboration among us, among our university. And Assalamualaikum warahmatullahi wabarakatuh. Thank you very much and wish you a very fruitful webinar today. Thank you very much. Pak Cahyo, thank you.
Thank you, Prof. Irma, for the speech and welcoming all the participants and the speakers. And the next, I would like to call to the, not to the stage, to the screen, Pak Dasa, Dr. Dasa, to deliver the opening speech representing the international program of IPB University. Dr. Dasa, the time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you, Dr. Cahyo. Thank you, Professor Irma, and also Dr. Asep. Thank you also to our international speaker for today, Professor Mehmet Ulassinar from Turkey, who has given the effort for today as well. Also, Muhammad Jasim, Mr. Muhammad Jasim, PhD from Bangladesh and now in University of Queensland. Also, medical doctor Aminul Islam, PhD from Bangladesh and now in Tokyo University. Of course, also thank you to our international speaker, also Dr. Asep Gunawan. Good morning to you all. The sun keep going rising. The cloud is hiding. Although the life is so confusing, COVID give us lesson and changing. Don't worry, this international webinar will make you signing. International speaker will be speaking. Information will be presenting and make your day interesting. Firstly, we would like to congratulate to Department of Animal Productions and Technology, Faculty of Animal Science at IPB University, who has successfully run the 5th webinar series and now international webinar, the 6th series of webinar for such creating amazing international webinar. The topic today is generation, handling, and publishing next generation sequencing research data. It is a bit difficult, but still needed in Indonesia especially. I am Dasa Hunayfi from International IPB University. Again, I would like to thanks the support from all the speakers, greatly appreciated, and highest appreciations to all. Professor Mehmet Ulassinar, Mr. Muhammad Jasim Udin, PhD, also medical doctor Aminul Islam, and of course, Dr. Asep Gunawan. Lastly, to open this international webinar, we wish that international webinar will give us inspire to learn genome data becoming better. Therefore, we should collaborate and inspire to publish data become faster to fulfill a reader desire. Let us enjoy and inspire as our life in sensor and have a nice webinar. Thank you.
Thank you very much, Pak Dasa. Thank you so much, and thank you also for the beautiful poem for starting the webinar this morning. Okay, I think everyone is on fire now. We can start the sessions. Thank you, Pak Dasa. I think I would love to. Of course, we would love to have you all along this webinar, but I believe I understand you other commitments in the office. So please feel free if you have other meeting or other commitments today. If you want to leave the sessions, just feel free, Pak Dasa. All right. Okay. Okay. Participants, I would like to start the first sessions of today talk by calling the first speaker of Dr. Asep Gunawan to the screen. But before he present his topic, allow me to briefly introduce the speakers. All right, let me share again. All right, okay. All right, I hope everyone can see my slide. Dr. Asep Gunawan is from Faculty of Animal Science at IPB University, particularly from Department of Animal Product and Technology. He's from Bandung, and education's background, graduated from Faculty of Animal Science, IPB University in 2003, and then earned his Master of Science from University of Bonn in Germany, and also for his PhD from the same university in 2013. He's a very prolific researcher in the department with the H index of 6 on the Scopus, and also with a Sinta score of 29.00 at currently. He also published already several academic books, as you can see here. It's in Bahasa Indonesia, Aplikasi Genetika Molekular, applications of molecular genetic in animal reproductions, and so on. And also has 2 research patents for the leptin receptor gene for selection fatty acid composition and cholesterol in sheep, and also the improvement of sheep meat flavor and other using candidate gene of CYP2A6. And he has published a lot of papers in Q1, Q2, and Q3 journal, more than 40 papers in PLOS, BMC Genomics, Animal Reproductions, and also the other journal, including Mid Science. And also he is recognized in many reputable journals as a reviewer, PLOS One Gene Reports, and also the other national journals. I think, Dr. Asep, if you are ready with your presentations, I will. The time is yours for presenting your topic. Thank you. Are you okay to start?
Okay. Okay, thank you, Dr. Cahyo. Can you hear my sound? Can you hear my sound? Yes,
yes, your sound is clear and like a crystal. Please.
Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Okay, thank you to the chair or moderator, Dr. Cahyo Budiman, for facilitate this international webinar and also introducing me to give the first talk. I would like to thank to Head of Department, Professor Dr. Irma Isnavia, and then Dase Hunayfi, Dr. Dase, for supporting this international webinar, to speaker, and also to participate, from different background, different university, professor, associate professor, student, guests from various ministry and privat and also government sector. So in this opportunity, allow me to share a little bit knowledge based on experience during working with RNA-seq research. In this talk, I try to elaborate the theory, practical work, and also experience related dealing with RNA-seq experiment. So that's why in this talk, I will give the topic, moment, sorry, it's a little bit, I have to share. So hopefully now is, can you see all the full screen, my screen? Dr. Cahyo?
Clear.
Yes, okay. So in this talk, I will try to elaborate the theory, practical work, experience related with RNA-seq experiment. So that's why my title of this talk is How to Perform RNA-seq Experiment. So the outline of the presentation is were divided into 7 parts. The first is introduction. Maybe some from all the participants is already common or familiar with RNA sequencing, but maybe some it is quite new. So that's why in the first we introduce what is RNA-seq, and then second, how to design the RNA experiment. This is maybe is more related with different kind background from all the participants who interest for this webinar. And then we will simplify 3 and 4, the experimental method and analysis method. And then after that, we will try to demonstrate the summary of data production. And then I will give brief also comprehensive study related with our research study, from the planning until the publishing data. And then the 7, I will close with a conclusion. Let's come to the first session. So at least on the beginning of 2000, around 2006 or 2008, or 14 or 12 years ago, RNA-seq is more become popular technology, especially in simple species or eukaryote species like plant model Arabidopsis thaliana, and then also bacteria, yeast, like that. But since 2010 until 2020, this currently research, this technology RNA-seq was applied in complex species such as a human and plant, and also in maybe it's related with our background is livestock. So before coming to the next deeply what kind of RNA-seq definition, so maybe I will give a brief introduction. RNA-seq is a method to measuring gene expression using deep sequencing. So maybe some of you know about the normal sequencing or Sanger sequencing. So this is more overcome the weakness from the normal sequencing. So the process is involved involved in reverse transcribing RNA to cDNA, using high throughput platform. So that's why later I will inform what kind of platform normally that we use. So the typical RNA-seq experiment aim to find differential express gene between two conditions. Actually, it's simple. We want to compare between 2 different phenotypes. I think in the reality or in the phenomena, we can have some big question. Why, for example, individual 1 and individual 2 is different. So we want to know the genetic mechanism, why is 2 different phenotype is different. So, and then the complete RNA-seq that we can get with other technologies can also complete to discover new transcript or splice variant, polymorphism, or some kind other gene expression. So here, like I mentioned in the previous slide, the typical RNA-seq experiment, to find differentially between two conditions. Yeah, for example, maybe the easy thing is between normal and disease. Yeah, between cancer and normal. So we want to see what kind of the genetic mechanism is causing different between two conditions, normal and cancer, for example. Yeah, this means when we want to know the different condition, this means we want to look at the different in gene expression. Yeah, so we can use RNA-seq to measure gene expressed in both conditions. Yeah, for example, in this case, is condition in normal colon and colon tumor, for example. Yeah, then we can figure out what is the difference. Yeah, after we have two different phenotypes and then we isolate the DNA and then we map to the reference sequence and then we can see what kind of the different thing. Yeah, which kind gene is active in the condition 1 and then which kind of gene non-active, for example, in the condition 2. Yeah, so I think it is more simple. Everybody can understand. So by this comparable, we want to know. Yeah, like that. So this is the basic principle of RNA-seq. Yeah, briefly, long RNA are first converted into library of cDNA fragment through either RNA fragmentation. Yeah, you can see this is the cut. Yeah, as a RNA fragmentation. This is the whole. And then sequencing, yeah, blue, this is the blue, yeah, are subsequently added to each fragment, yeah, a short sequence obtained each using high throughput sequencing technology. So this is the fragment of RNA, and then with the help from the adapter or maybe the sequencing will attach, yeah, to the RNA fragment, yeah, and then the resulting of sequence read, yeah, are aligned. This is if we see this is like a book, yeah, reference, yeah, are aligned. This is our sample, yeah, are aligned with the reference genome. This is the reference genome, yeah, or transcriptome. Then we can get 3 classified. Yeah, the first result is exonic. Yeah, this is the exonic. Yeah, like that. Exon part, it means maybe you know this is the coding region that important because associated with the protein translation. And then also the second is junction read, yeah, like a splicing. Why between one individual and other individual, maybe it could be different, it could be the same gene but different variant. Yeah, and then the third is poly and read. So this is actually the basic principle of RNA sequence. So RNA-seq, what kind of identification that we can analyze from the RNA sequencing? Yeah, so RNA-seq can identify more comprehensive analysis, yeah, such as genome resequencing, yeah, and then this is single nucleotide polymorphism. Yeah, normally when we do approach a single gene model, we use or we characterize single nucleotide polymorphism. Yeah, but with this RNA-seq, we can also simultaneously get the whole, yeah, SNP in coding region. And then epigenetic event, yeah, this is also maybe now is we are become more popular also to come this epigenetic event, and also alternative splicing, and then small RNA identification, yeah, like microRNA, yeah, maybe otherwise it is not coming yet in our research work, but maybe later it will be become more popular. And then the most popular when we read paper or publication related with RNA sequencing is to capture the transcriptome profile, yeah, like in different kind of phenotype or some kind of the tissue, yeah, like that. Okay, so this is that I highlight the transcriptome analysis. Yeah, so commonly, or yeah, like I mentioned before, when we read the paper or publication, RNA-seq were performed to identify transcriptome profile. Yeah, but what actually the transcriptome? transcriptome related with the sign, maybe, or related with the part, but some of you also here maybe like a transcriptomic, transcriptomic. When we mention about transcriptomic, maybe it is related with study. So what actually the transcriptome is the complete set transcript in a cell and their quantity for a specific development stage or physiological condition. So we want to know the transcript, in the cell, like in muscle, in liver, or maybe in other specific tissue, which two different physiological condition, in specific developmental also. So what is the important transcriptome? By transcriptome analysis, we can interpreting the functional element of the genome. We can see, for example, the functional, either in biological function, molecular function, or cellular function. And then second, to revealing the molecular constituent of cell or tissue. The expression is highly expressed in specific tissue. For example, in liver, in brain, it is also related with expression. And then the third is understanding differential expressed gene between two conditions. We can capture, we can highlight what is the substantial element can make a differentiate between the two conditions. And then after we know the basic principle of RNA-seq, so if you want to conduct research using RNA-seq, what is the important? What is the important step and how to design RNA-seq experiment? So that's why this is the 4 question that you have to answer. Or maybe you have to make hypothesis. Before we start the RNA experiment. Because this is also related with the cost. It's expensive maybe if we compare with the normal sequencing. Some question that we have to address. The first is, why do we expect to find differential expressed gene in particular? So maybe this is related with what kind of the important. If we, for example, want to know the between normal and the cancer, it is very clear. So maybe by know the differential between normal and cancer, maybe people can can make a other strategy, how to treatment, how to make a treatment, how to make a like a pharmaceutical or something like that. This is maybe too easy to make easy. And then what type of gene that you expect to find differential express? Some of us maybe know about when we want to compare between two different conditions, maybe we know what kind of gene that have a function. Like for example, when we compare between normal and Alzheimer's disease, for example. So maybe we can expect the gene which is expressed in the brain, or maybe in hormonal, it could be expressed. But we didn't know which kind of gene, it is related with Alzheimer's, for example, with Alzheimer's disease, whether apolipoprotein or something like that. But at least from the read or the single gene, study, we can know the function of gene. But maybe in our sample we didn't know, so that's why we want to know the differential express. And then what are the source variability from your sample? So this is the crucial that you have to really take care, your sample that you want to compare. Don't our sample should be uniform and not many other environment factor, because it could be make a bias. And then where do we expect most your variation to come from? So you have to make sure, our sample, it should be the same. And if you know the variation come the environment, maybe it could be make a bias. So let's talk if we learn about the molecular approach to understanding complex thread. So now we talk about maybe our background, could be livestock or maybe plant. Yeah, in this regard, for example, I give example meat quality trait. Yeah, it is very complex trait. Yeah, some molecular approach try to identify candidate gene. Yeah, only 1 gene, 2 gene, or something like that. Or maybe they try to find out the position in the chromosome, which is important to control this trait. Some other also now to identify using genome-wide association study to make a combination between SNP, single nucleotide polymorphism, and the position of the chromosome. Yeah, but with this next generation sequencing, especially RNA-seq, yeah, we can get more combination analysis, such as candidate gene. We can get also the position of the chromosome. Also, we can find out, and also the gene expression that normally can do by qRT-PCR. Yeah, by this RNA-seq we can identify it also simultaneously. Okay, so related, maybe this is the more illustration, yeah, more clear illustration. Yeah, for example, related with meat quality. So we try to identify using RNA deep sequencing, yeah, related with Borten. Yeah, Borten it is unpleasant odor, yeah, in meat, yeah, especially in pig, yeah. Because this work in Europe, they consume mostly pig meat, so they have a problem with this. Yeah, that called Borten, unpleasant odor. Yeah, so unpleasant odor due to by two chemical compounds. The first is androstenone, yeah, related they produce in the testes and also in liver, and then the second is scatol. Yeah, normally they have a catabolism or metabolism in the liver. So in this problem, why this is become problem? Yeah, because people, yeah, normally they doing castrated, yeah, from the beginning, yeah, when 7 days, for example, to avoid, yeah, to reduce the Borten, yeah. But in Europe they have a commitment, yeah, or maybe they have a consortium, yeah. In 2018, the castration will be banned, yeah, will be banned, yeah. Maybe other alternative they have like a vaccination, but maybe it's costly and also maybe it's not really what is practices. So that's why, because this androstenone and scatol is more genetic influence, yeah, with the heritability is high, yeah. So that's why they try to identify the genetic factor which is control these two components. Yeah. So this is the simplified the experiment that we create. Yeah. So we measure from 100 boar. Yeah, boar is a bull of the pig. Yeah, and then we slaughter this boar, and then out of 100 we select 10 boar with represent extreme high and extreme low Borten. Yeah, so from that, and then we measure the source of the Borten become 2 from testis and liver. Yeah, so we isolate both kind of this tissue, and then we measure, yeah, the androstenone level. Yeah, so from the 100, and then we get, yeah, extreme 5 with the mean 2.48, and then the low, and then we make a categorize 0.25. Yeah, so we have 100 animal out of 100, and then we choose which one is represent the divergent phenotype. So we take the top up high 5 and we took the lower 5. So maybe this is not like a different condition like normal and disease. Yeah, this is quantitative trait, is little bit tricky. So that's why we have to manage it. Yeah, so from both tissue, and then we do RNA isolation and quality control. So the second example also that we already done, we use RNA-seq to identify candidate gene for selection meat with high unsaturated fatty acid in chicken. So this is related with the functional food. Yeah, maybe people now is wondering to eat meat, yeah, because associate with some kind of negative impact. This is maybe it could be totally true, or maybe it's a hypothesis. Yeah, like they have a degenerative disease, cardiovascular or something like that. So that's why in this regard, yeah, we identified, yeah, meat, yeah, and then we measure the fatty acid composition. We took 62 animal, yeah, a chicken, and then out of 62, and then we measure the fatty acid composition, yeah. So from that, from that, and then we can classify, yeah, we can classify, because our goal is to differentiate between unsaturated fatty acid, and then we get, yeah, we get the average is 54, and then we can classify the high is 57.06, and then the low is 51 point something like that. So by this, maybe in the sixth part, I will give briefly comprehensive study from our house, like that. Okay, so this is also the typical, and then so the experimental. So now this the third part. There are 3 main steps for RNA-seq experiment. Yeah, the first is prepare a sequence library. This is the crucial preparation, because good or bad the quality of RNA-seq depend on how to prepare the sequence library. And then the second is sequence, and then the third is data analysis. Maybe Dr. Amin will help us how to analyze, and will demonstrate how to analyze this data. So the first is workflow is isolate from the total RNA from the sample interest, like a liver or maybe testis, and then eliminate the DNA contaminant using DNase, yeah, because we work with RNA, yeah, and then choose appropriate kit library preparation, and then we will make a randomly fragment, yeah, purified for short read sequencing, and then reverse transcript fragmentation into cDNA, and then we use also ligase adapter, yeah, both to cDNA fragmentation, and then we get finally the product size, yeah, between 200 until 14 base pair, yeah, like that. So out of this product below 200 or 36, it will be clean, yeah, it will be clean. I more emphasize to the sample preparation, yeah, and then to the second library construction. This is very crucial, yeah, because good or bad the quality of sequencing is depend on the preparation, yeah. So what kind of the simple way that we can provide, yeah, to isolate the RNA, yeah. So this is, for example, you have a tissue that you want to compare, like a liver, yeah, and then we isolate, yeah, the RNA, of course, using a kit, and then the simple way to check quality RNA, yeah, using RNA after RNA excision using gene electrophoresis, yeah. So the good quality could be seen, yeah, by showing 2 ribosomal RNA, yeah, 18S and 28, yeah. So this is the thing, that simple way that we can measure. If you find the product is like this, is like that, so maybe it's good, yeah, it's more good, yeah, when you do analyze more quantitatively, yeah, like using a spectrophotometer or maybe bioanalyzer, yeah. But make sure from the first your quality RNA is good, like that, yeah. So if it is okay, and then you can continue, because this is quite expensive. Maybe you can measure quantitatively, yeah, more accurately using bioanalyzer, yeah. So if we make a quantitative from dead to band, yeah, the gel electrophoresis, you can see, yeah, the represent a good quality of RNA is RIN, RNA integrity number, yeah. The score is can start from 0 until 10. Yeah, 0 is bad, and then 10 is good. Yeah. So normally for RNA sequencing is required, yeah, greater than or equal 7. Yeah, 7. Yeah. RIN equal 6. Yeah, sometimes they hold. I mean, they require to repeat, but it's also working if you want to continue for sequencing. But below than 6, yeah, I suggest to recommend it. Try to repeat again. Yeah, they have a critical work, yeah, to isolate the RNA, especially maybe with liquid work, yeah, like semen, yeah, or maybe milk or other like that. Yeah. Okay, so this is, for example, that we see, yeah, from our sample. Maybe it is not like 10, yeah, not 10, yeah, but maybe above 6 you can predict, yeah, because we can see they have a 2 band, yeah. You can see before they have a noise, yeah, here, but here is very clear too. But in here, maybe we can expect our RNA is more than 6, yeah, more than 6, because only, yeah, they have a little bit noisy, yeah. So this is then that when we analyze by bioanalyzer. So this is from a pilot study that we measure our RNA, yeah, and then we can see, yeah, this is actually after 3 or 4 experience, yeah. The first experience, yeah, maybe, yeah, 70% is fail, yeah. But after we know the protocol, and then we can see, yeah. So when we isolate the first RNA, and then the result is good, like that. So this is the after third time, and then we can see the result like this, yeah. This sample is 9.9. It means like absolutely perfect, yeah. But other, yeah, I think above 7, yeah, except this 7.8 and 7.3, yeah. But this is whole. It means if you have 3 replication biology or 4 replication by 5 replication technical replication, it's better to continue like that, yeah. So, and then after sample preparation, and then the important thing also, we get the sample preparation, and then library, and then sequencing, and then we have a raw data, yeah. So from the raw data, we have to make also the quality control, yeah, QC, yeah, what kind of parameter that we have to consider, yeah. And then after that, we have a pre-processing, and then our sample, because our sample is small, from 200 until 400 base pair, will be alignment to the reference sequence. Yeah, for example, if you work with a chicken, maybe you have to download the reference sequence, for example, Gallus. And then sheep, maybe you have to download the new version Opis aries. Or maybe, if you work with cattle, maybe you can download the Bos Taurus, like that. And then after you map, you get the expression, yeah, between 2 conditions, and then you can compare it, and then you can do enrichment analysis, yeah, which kind of functional categorize, yeah, pathway analysis and something like that. So this is that I explained, yeah, from that explanation, yeah, from the preparation until enrichment analysis. So, and then the 5, yeah, after sequencing, and then you get the summary of the data production, yeah, of the data production. So there are 4 important parameters, yeah, to solve the summary of data production. The first is raw data statistic, and then the second is average base quality per each cycle, and then trimming data statistic, and then average base quality each cycle after trimming. Yeah, like we have to really make a clean. Yeah, we have to make a clean from the noisy. Yeah, to make sure our sample, yeah, can more perfect align like that. Yeah, for example, this is the raw data statistic that we get. Yeah, this is the some parameter or indicator we have to see. Yeah, the sample from the GC, yeah, GC percentage. Maybe if you order some kind of primer, yeah, one gene, and then you order primer, you have to consider the GC content. Yeah, not to be far, yeah, between GC, maybe 45, range 45 until 50, maybe it's okay. But if some 30 and some 70, maybe it's quite wide like that. Yeah, so we can see the average of GC content, yeah, is 45. And then they also some kind other parameter 80 and also QC, yeah, quality 20 and 30. Yeah, so this, for example, in quality 20, yeah, represent if they have 100, yeah, nucleotide, maybe one it could be error, yeah, 94. But in quality 30, yeah, it means 1 out 1,000. Yeah, if you have 100 nucleotide, yeah, one could be error, yeah, or one could be maybe it is not working. So that's why for RNA-seq, it's better to use. Normally, we use the quality 30, yeah, because to avoid the bias. Yeah, you can see whole the genome, and then we want to make a alignment. Yeah, if maybe we use 20 or 100, maybe it's become false. Yeah, but if 1,000, maybe we can minimize like that. Yeah, so this is the indicator of base quality each cycle. Yeah, this is you can get the information from the bioinformatica Abraham. So this is the represent, yeah, how our quality of sample, yeah, our quality of sample. Yeah, the green background is good quality. If you have the sample, yeah, in this area green, it means your quality, your sample is perfect. Yeah, it is mean you have no need to treatment anything. But they also some orange, yeah, orange background is acceptable quality. I mean 20 is also okay, yeah, but normally we use 30, yeah. And then red background is bad quality. This, yeah, if 10Q, yeah, 10Q, it means, yeah, out of 10, yeah, nucleotide you have one error path. Yeah, this is the interpretation, yeah, like that. Yeah, so it means this sample, what kind of the sample? This is good or bad? So from this mapping, we can see, oh, this is not good sample. Yeah, because they distribute in 3 zones. Yeah, green, yellow, and also red. This, for example, this is our sample. Yeah, when we see, yeah, the first, yeah, our sample is distributed like that. Yeah, yeah, it is not good. Yeah, actually. So it means we have to repeat again. Yeah, like that. Yeah. So this is distribute in 3 parts. Yeah. But when you see here, yeah, this is good for base sequence quality. Yeah. Yeah, they have this, yeah, some noisy, yeah, but still acceptable because in 20. But this also we can treatment. Yeah, that's why maybe I mentioned before is trim. Yeah, trim to clean, yeah, to clean the files discovery like that. Yeah, so you can now differentiate between good and bad base sequence quality based on FAIR quality. Yeah, 10, 20, or 30. Yeah, so this, if you have a look, yeah, trimming data. What is the trimming data? Yeah, the trimming data, yeah, program is used to remove, yeah, adapter, yeah, base quality, yeah, base quality lower than 3 from the end. So it means they have still, because the cDNA they attached by adapter, maybe the adapter still attached to the cDNA. So that's why we have to do the trim, yeah, to remove, yeah, the adapter. Yeah. So, and also from the trim, they can remove, yeah, shorten base, yeah, below 100. You know, our product is 200 until 400 base RNA fragmentation, and then below, for example, 36 base pair are dropped to produce clean data. Yeah, below that they suddenly to make a clean because it's noisy. Yeah, like that. So you can see here before trimming they have, but after trimming, yeah, so you can minimize this. Yeah, you can minimize it. Yeah, all now is in good quality. Yeah. So raw and data versus stream data. So after, for example, we have a raw data 4.4 GB, and then after we trim, so maybe it's become reduced. Yeah, but the data quality is good. Yeah, the data quality is good. Yeah, like that. So the analyze, yeah, after we trim the data, now our data is clean, and then we can see also the good quality. So now the thing is how to alignment our sample to the reference sequence. Yeah. So now, like we have a book, we need like a scanning. Yeah, we will make a copy, yeah, to the scanning. Yeah. So from this also we will align our sample to the reference genome. Yeah, to the reference genome. So for example, this we have a data, yeah, from all the sample, they can alignment, yeah, with the range 89 until 91, I think the high. Yeah, so maybe it could be acceptable. Yeah, the first experiment, yeah, in 2008, when people firstly doing RNA-seq, yeah, the percentage of map, yeah, is still lower actually. Yeah, 45 until 50 is still acceptable and they can publish. Yeah, but now I think because they also advance, they they improve also the reference sequence, and then our equipment or maybe facility protocol maybe already established. So I think now is quite close to 100. Yeah, like that. So it's more better now. Yeah, you can see from our result.
Hello, Dr. Zeb.
Yeah, and then so now maybe I will speak about the application of RNA-seq. Yeah, application RNA-seq in livestock were demonstrated in several traits. Yeah, here and also in different species. Yeah? If before maybe working with eukaryote, now is already, yeah, like a pig, because it's especially for the economically important trait. Yeah, like a meat quality. Yeah, like a fatty acid composition, a marbling, yeah, or maybe other. Yeah, they can invest a lot of money, but maybe they can get the comprehensive analysis. Yeah, like this. Yeah. So let's come to our case. Maybe I will try to give comprehensive, yeah, from the planning what we are plan, and then we try to provide what kind of the expected result that we get. Yeah. So the problem is maybe in Indonesia or maybe in I don't know, in Middle East, maybe they didn't have a problem with the meat quality of sheep, but maybe Indonesia they have, yeah, some specific gender like a woman, they didn't like the sheep meat because they have unpleasant flavor and odor, or maybe bau perengus lah kalau kita bahasa Indonesia kan, ya. So then we identified the unpleasant flavor and odor in sheep is due to by branch chain fatty acid. Yeah, branch chain fatty acid, they have
excuse Mr. Asep.
Yeah.
Excuse. Sorry, Mr. Asep, time is up.
Yeah. Okay, a little bit.
Thank you.
So, and then from this, we try to identify, yeah, the sheep meat odor. Okay, so this is the same. Yeah, we find out from 3M high and low, and this is that we create. Yeah, what did we expect is differential express gene and SNP. Yeah, so this is the map. Maybe it's not good compared to 2009, but this is okay. And then we get the differential express. Maybe Dr. Aminul will expand later. Yeah. So this also functional categorized, and then we can get also the single nucleotide polymorphism. Yeah. And we get the strong candidate gene, yeah, after we validate it with qRT-PCR. Yeah. So, and then later on, if you interest to use this as a marker, assisted selection, you can verify it, yeah, in your population, yeah, polymorphism, and try to associate with the trait. Yeah, for example, this we can identify that maybe is not high, but we can know, yeah, which genotype is low sheep meat odor and flavor.
Yeah, for example, AA genotype. And then finally, I will close with the conclusion. The RNA-seq experiment produces higher dimensional data. And then in order to perform a robust statistical analysis, the methodology must be carefully designed. Yeah, you have to carefully. And then RNA-seq also provide the resolution transcriptome study, yeah, gene expression, and also gene annotation. And then the strong of RNA sequencing can identify, yeah, other phenomena like a transcript, splice variant, and also single nucleotide polymorphism simultaneously. So that's my talk. Sorry, I interrupt the time up, but hope maybe it can make a little bit description when we want to dealing with RNA sequencing. So thank you. I close. Assalamualaikum warahmatullah, and I give back to the moderator, Dr. Cahyo. Thank you, Dr. Cahyo.
Thank you, Dr. Asif, for the talk. I hope my voice is clear enough. Okay. All right. We have not so much time left. I would like to call the second speakers, Associate Professor Dr. Muhammad Aminul Islam, and I believe he's already on the screen. Dr. Aminul Islam, are you there? Hello? Hello? Hello?
Can you hear me? I'm there.
Yeah, okay, all right, okay. So before you start, I would like to brief you, share your CV to the all audience. Give me a few seconds, Doctor.
Yes, please.
All right. Okay. The second speaker is from Department of Medicine, Faculty of Veterinary Science, Bangladesh Agricultural University, Muhammad MD Aminul Islam, DFM, MS, PhD. He born in Bangladesh and then now working in Department of Medicine, Faculty of Veterinary Science, Bangladesh Agricultural University as Associate Professor. For the education background, he got his DFM, Doctor of Veterinary Medicine in Faculty of Veterinary Science from Bangladesh Agricultural University, SAM University also from his MSc, obtained on 2009, and got the SAM PhD from SAM University with Dr. Asep and University of Bonn in 2017. He's very prolific researcher, publishing a lot of paper, which X-index citations more than 300, H-index more than 10 in the Google Scholar, and also received so many prestigious awards, including FEMS Congress Attendance Grant, Best Publications Award, GSPS Fellowship, Postdoctoral Research, which already completed since last year in Tohoku University, Faculty Prize in 2017, and also Young Scientist Best Presentation Award in 2012, and other awards that he already received. He published more than 31 original research article, more than 42 conference abstract, and other publications in newspaper, academic dissertations, and also others. Also recognized as in many others, in various international journals, sections editor in Bangladesh Journal of Veterinary Medicine, associate editor in a Journal of Advanced Veterinary and Animal Research, and also review editor in Veterinary Science. And to note also, he is a reviewer from on the some Q1, Q2 journals, PLOS ONE, Frontier in Immunology, Gene, BMC Genomics, Animal Science Journal, and also in ACTA Veterinary Science Scandinavia. I think I'm very glad to have you here, Doctor. And then I think time is yours to present. Hopefully you can deliver your speech in 30 minutes or 40 minutes before we end up with the Q&A session. Thank you.
Thank you, Mr. Moderator. Very good morning to you all. 3 weeks ago, I had a conversation with with my friend, Dr. Asif Gunawan. He was telling me that they are going to organize a virtual meeting, and it's about transcriptomic research practice, and asked me whether I could able to conduct a sessions over there, and I gladly accepted, and I'm very happy to be here too with you all. So what you told about me is perfectly fine. So I'm not going to introduce myself again. So what about the topics is I'm going to talk about. Let me have my screen shared to you. Yeah. I'm going to talk about some tips and techniques for bioinformatic analysis of transcriptomic data that could obtain from RNA sequencing or microarray, whatever. So what is bioinformatics? You know, it's in 1970, Pauline Hogberg and Ben Hespers, Dutch scientist, theoretical biologists and mathematicians, they just for the first time proposed a term bioinformatics. As a definition, this is the study of bioinformatic process in biotic system. So it's a combined or integrated disciplines. It includes chemistry, biochemistry, biology, statistics, mathematics, engineering, computer science, and so many other disciplines altogether. If you simplify it a little bit, it comes the statistics, biology, and computer science are core subjects that integrate this information science we call bioinformatics. So why should we need the bioinformatics stuffs? If you say simply, it's if you say simply a quick answer, it's for biological interpretations of transcriptomic data. If you think about a whole transcriptomes for a mammalian genome, it's a deal about 25,000, more or less 25,000 genes that could be analyzed by microarray or next generation sequencing. So it's a huge amount of data. So after different statistical modelings, we could come to narrow a little bit down about, say, for example, 100 to thousands differentially expressed genes in response to a conditions or developmental status, whatever the treatment given. And then it comes a little bit narrowed down, say, for example, 100 to thousands. Still, it's a big one. Then we are thinking or we are focusing about a key or candidate genes. That could be a very short-listed one, 5, 10, or 2, whatever. And not only that, these short-listed genes, we are also interested what are those genes and what they do in the biological systems. So all these things we could know by the implementations or incorporations with the bioinformatic knowledge. So transcriptome is a big thing. As the first expressions of genomics, if we think about the central dogma of molecular genetics, it comes from the DNA to RNA. It's a transcription process, and different types of RNA are produced. And particularly the messenger RNA, that one encoded with the protein that ultimately terminated with protein expressions that control the phenotypes of particular interest. And some other RNAs are there, transport RNA, ribosomal RNA, also some sort of non-coding RNAs we call microRNA. So altogether, this RNA family we call transcriptomes, though our focus mainly on the messenger RNA that sometimes we frequently call it gene. Then the gene who is encoded with protein that ultimately express the protein and particularly control the phenotypes. So for this transcriptome or transcriptomic study, there are different approaches to get the transcriptomic data. Dr. Asif was talking about the RNA sequencing in details. I'm just show you a little bit comparative or comparisons between microarray and RNA sequencing, how they could implement for getting the transcriptomic informations. Microarray, it's a little bit back or earlier approach, and RNA-seq is a latest approach. But ultimately, what they do is they give us a bunch or list of differentially expressed genes. But what they actually do in between, for example, if we start with transcriptome profiling things, we have to prepare the total RNA samples from our targeted samples or as a control samples. And this targeted and control samples, total RNA samples, we go either by microarray or by RNA sequencing. If we choose microarray, it's a hybridization based approach, and we need to color or label our total RNA samples with a fluorescent dye. Then we need to hybridize these samples in a gene chip or hybridize array techniques. And ultimately, we need to clean it and wash it, and after finally scanning these chips, and we got the image file, that is image intensity data. On the other hand, for RNA sequencing technology, it works with sequence-based techniques. Each and every fragments of the RNA having in the samples, it amplifies and sequence each and every RNAs. And then finally, we can count the sequence as a short read. So how the data or raw data looks like? The raw data come from the microarray techniques. It's just an image data. We converted it to a numeric value as an intensity, image intensity. On the other hand, RNA sequencing data, we can get count data. It's a short sequence read. Then for this microarray, raw intensity data, we need to do some sort of background corrections, normalizations, and then summarizations of this data to get the clean, ready to analyze raw data, or process data, we call. And that is the continuous data or continuous distribution, data having continuous distribution. On the other hand, the RNA sequencing, we got the short read, and we need to align this short sequence to a reference genomes. Then we can summarize expression data as a count data. And in addition, this count expression values, we can get the novel, that is unknown gene. We can also have the information about it from RNA sequencing. That is unlikely from the microarray techniques, where the only fixed probe we can design, and we can get whether this probe is hybridized or not. And then this downstream statistical analysis from both sides, both data set, we could get a list of differentially expressed genes or transcript. These differentially expressed genes or transcript is the raw materials or basic input for the subsequent functional or bioinformatic analysis, what I am going to talk about in detail. The bioinformatic analysis is a different way or different pipelines, but more or less the basic principles is we need to, what I said first, that what bioinformatic analysis is the things we do to biologically interpret our data, what the data says about the biological phenomena. That's why we start with the short-listed differentially expressed genes. We got it some sort of criteria selections and what things. Then we need to know what the biological phenomena represented by this gene. for these things, two major categories of the functional analysis we need to do. One is gene ontology analysis, and another is functional pathway analysis, or sometimes latest we call this network analysis. Previously, only the concept was pathway analysis, but later nowadays it's a little bit advanced approach for network analysis. So for gene ontology or geoanalysis, it gives us three types of informations. One is molecular functions, particularly what the molecular functions, say for example, it's a catalyst or what else is caused by the differentially expressed genes. Then what cellular components, I mean this, which part of cell it works with. Then it comes with biological process, that is the most important one. What the biological mechanisms they control, something like this is the biological process enriched by the differentially expressed genes. After getting the geo impressions, we have some sort of impressions of these data sets. Okay, this data have this kind of biological phenomena could be involved with, and we could go back our experimental design and make a cross validations. Okay, what was the primary intention and what it gives us. Then after one cross checks, we could step forward for pathway analysis. For the pathway analysis, there are until now there are three kind of approaches have been practicing. One is over-representation analysis. It's a bit earlier one that I'm going to talk about. And then second one is gene set enrichment analysis. And the third one is topology-based network enrichment analysis. So for the over-representation analysis, or as a whole, how it works. For example, initially we got the, this is a symbolic picture of microarray, but RNA-seq also similar types of phenomena. What I said, the initial product is differentially expressed genes. So for this differentially expressed genes, once we get this DEG, differentially expressed gene list, we need to choose a database from where we can compare our gene list. And once the database is chosen and our DEG list is in hand, then we could go forward either over-representation analysis or functional class scoring or pathway analysis. Any sorts of analysis we can go, but one by one. Better first to go for over-representation analysis. What I said, it's a bit earlier, and what the way it works is from the differentially expressed gene, we use a database where each and every pathway has a list of genes already curated and experimentally validated, and we give our gene list, and from our gene list how much genes are overlapped with the existing gene list of particular pathways, and then it did a statistical test whether it's significantly over represented or not. Then the result comes as a time count, the pathway names, and the time number count, and whether the p-value is significant or not. Then the second one is functional class scoring. In this case, we did not know the particular pathway and their gene list in the database, but we compare our whole gene list with a very big one, big unclassified gene list, and we find out a certain list of genes that could incorporate with a particular pathways or biological functions. This we simply called gene set enrichment analysis. And there are a lot of open source tools for these things also. Then the next one is pathway or topology based analysis. Sorry. Topology based analysis relies on the genes interactions among them or the neighboring genes as well. For example, if we have 100 genes in our gene list, these 100 genes, how they interact among themselves, and also the similar sorts of genes, predicted genes from the database, and ultimately what sorts of biological phenomena they could control. This interactive networks approaches is recently getting popular. There are a lot of open source tools for this as well, and there are many sorts of analysis options are there. For example, protein-protein interaction, gene-gene interaction, gene microRNA interaction, gene transcription factor interaction, gene disease interaction, so many. So overrepresentation analysis, what I already probably told about. Then overrepresentation, we can sort out, or we can fix some sort of threshold. For example, p value, or sometimes we need to do a multiple test corrections after the representation analysis. Then we fixed threshold value for false discovery rate about 5% or near about. And this is the one kind of criteria we should fix to choose what the pathway significantly overrepresented by our differentially expressed genes. Then each pathway input genes that are part of our counted genes in this process, how many times they are repeated, and the background list genes. And the next, ultimately, every genes are tested over or underrepresented, then the pathways is up or down, and significance tested by some sort of statistical test like hypergeometric test, chi-square test, or binomial distribution, and so on. So many open source tools for this sorts of analysis. I mentioned here one is InnatedDB. It's a very relevant and very rich one, particularly in my and in our field. There is innate immunity related stuffs, but so many others related tools also available online. You need to choose which one is best fitted for your data set. What the data, I mean, what the fields are you are working with, what's your data about, and you can choose the best one for your data. Then this process has some sort of limitations. What I told before, that this overrepresentation analysis test the number of genes, but ignore the values associated with genes. For example, we just pick the differentially expressed genes list only, not like how the intensity or how their fold change is, either 5, 7, or 1, or 10, or -2. These sorts of informations are ignored in this process. This is a very big limitations of this approach. And second, it typically considers only the significant genes. But the concept is coming, that co-expressions and other things, that not only significant gene or highly significant gene, if a lot of genes have a little significance or only a little fold change, maybe they didn't pass the statistical threshold value, but still they have some sort of trend, either upregulated trend or downregulated trend. These sorts of genes could combinedly also influence the biological phenomena. But in this ORA approach, this little or few or insignificant up or down regulated genes are missing. That's two major limitations of these approaches. Then with these limitations, to overcome these sorts of limitations, different approaches are coming, but still the ORA is continuing. But the next approach, what I call gene set enrichment analysis. It works with a functional class and it considers all the genes of our expression data set, not the differentially expressed genes. This method works, or this method starts with the expression data list. What I show before, the microarray data set, it comes as intensity values, whereas RNA-seq, it gives us a count raw read. From this, from that raw data, it works with this functional class scoring. And for this also a lot of open source tools are available, but I mentioned here one. This is GSEA, the name is typically one. It's from Broad Institute, USA. And this method comes, and this method can overcome the limitations have ORA method, I'm sorry. Because this method don't require any threshold value. Rather, it works with all the gene list. And when this ORA ignores the molecular measurement or identify the significant pathways, but this FCS method use the information in order to detect coordinated change, not only particular short list of differentially expressed genes. It consider all the genes what up-regulated and down-regulated, just only two categories, irrespective of their fold chains, irrespective of their significance level. Then by considering the coordinated change in gene expressions, it accounts the dependence between genes in a pathway. The genes are coordinately and dependent to each other to control a function. That is the main issue addressed in this method, but in ORA method, it was completely ignored. Then the next, the very recent approach came. It's we call it a network approach, and it based on topology or the topological situations or status of the gene interacting with each other. Once the interaction things comes in more concentration, this the GSEA systems, and in this topology based network analysis, this interaction stuff got more priority, I would say the most priority, because a biological functions is a result of many interacting molecules, cannot attributed by just a single molecules, whatever the significance it is. Whereas the ORA or previous two methods completely ignores these things, and the individual pathway they considered as an independent way, not dependent or interactive or coordinated way. That is the advantage, or that is the latest add-on for this network-based analysis. Therefore, this network topology-based interacting network approach is much better compared to the earlier one. So what about the three techniques for functional analysis or pathway analysis I talked about? You need to prepare your data and you need to select a best tool for you, just as a preparatory task. So for data preparation, with the scientists from animal science background or other life science background. And this gene or molecular genetics database for particularly for human being or some sort of laboratory animal species like mouse or rabbit, these are very much rich and well annotated. Whereas for cattle, sheep, goat, pig, other livestock species are not that much well annotated. That's why the informatic or bioinformatic depth is not that much compared to the human genome informations. That's why if we want to get a better insight of our data, maybe come from sheep or come from pig or whatever other livestock species, we need to convert our data or convert our differentially expressed genes identifier to a compatible one that is particularly compatible with this particular tool. In most cases, it's better way to go for the human orthologous identifier if otherwise not happen. Then it comes a matter which tools I need to choose. A lot of things you need to consider for this, particularly the which database the tool is linked to it, and how much resources or data, in our case a gene, is curated for this database, how rich the database is, and what the method they follow for curating the database. Are the genes are only literature curated or they are experimentally validated? These sorts of things you need to check. And what the tools offer you, how visual outcomes or visual display figure quality you have. And finally, it's also you should consider that how easy to operate these tools. Being a biologist, not a computer people or statistician, it's a little bit tough job to handle these sorts of omics tools. So some sort of tools are very much user friendly, some are not. So you need to consider that one also.
Hello, doctor. Sorry to interrupt.
Yes, please.
You have less than 5 minutes to end up. Can you, would you mind to speed up?
Okay. I think I'm about to do my talk, but anyway, once your data is prepared and you choose your desired tool, then you need to go for, and there are particular tools have particular tutorial. You can find it out, because I'm always talking about the open source one. Then everywhere there is a tutorial, but you need to think in mind that if you compare with the licensed one, the open source tools are always a little bit tricky to use, not so user friendly compared to the licensed one. So you have to take some sort of time to learn it up, then you can go forward. Here are some very popular, I would say, popular open source tools for the bioinformatic analysis. InnateDB, I mentioned last, then Geo, Debit, Cake, GSEA, Reactome, Pathway Commons, Pathway Tools, and PathPGeo. And here are the links. You can find it out later if you're interested in, and you can find the particular tutorial to go for. I have a plan to go in detail, I don't know, can I have 10 minutes time more?
So maybe 5 minutes should be okay, doctor.
Okay, then there are, I have a plan to show, a short demonstration about pathway analysis and the network analysis. Then say, for example, better I can go for this two demonstration one by one. This InnateDB. For the InnateDB, using this InnateDB, if we have a differentially expressed genes, I have a data from my own experiment. I just copy it from this. It is about innate immunity of pig after a vaccine trial. some sort of things. That's why I just choose this InnateDB database. And if I go for pathway analysis, then I need to follow this waveform, and there is a space you can find. You can just paste your gene list and do it upload. Then the window comes and you need to check it out, and you need to identify your data set. I use, it's a cross-reference data because I use the human orthologous identifier here, and it's ensemble identifier I use because my data came from porcine genome. The porcine annotated file is not available in this database. That's why I shifted to human genome and converted my data to human orthologous ensemble. Then next, it comes, I would say it's ORA based pathway analysis. I can just click, then some sort of parameters I need to choose. I need to choose an algorithm here. It's common and it's recommended here. Then I can just do the analysis.
We cannot see your screen. We cannot see your demo here. We just only see your PowerPoint.
Maybe
you can turn off your slideshow of your PowerPoint and then share again your software that you want to demo.
Yes, sorry.
Okay, no problem.
Can you see now?
Not yet. It's still on your PowerPoint. I think it's somehow it's freeze.
You share.
Okay.
I think.
Okay, we can see here. All right.
Okay.
Perfect. All right.
So it's better I go a little bit back, just a minute. So what I told you that I have uploaded my data and I step forward with the instructions here. Then it gives me a list of pathway overrepresented here, and after overrepresentation, I did a multiple test correction by HG correction method, and after this, finally, I got this file or figure. There are Excel file attached to it, associated to it, and I can download it in MXL, Excel, or Tab, or CSV. This is okay, and this is based on Cake database. I can ship to database on Reactome. I can ship to other database. Also, I can choose all sorts of database together, and then it gives some sort of this way. And this is the threshold value. It's a recommended significant threshold is 1.3. It's recommended by the program. So this is the blue line, the threshold value. That means the bar has up to upper, then the blue line is significant one. I can see a little bit here also. That means a lot of the pathways are overrepresented with this gene list. And we can do another analysis also from this uploaded data. For example, gene ontology analysis, I can do it here also. You can see again the way I followed last time. This is just cross reference, and this is ensemble. I chose because my data is from human one. Then this is the ontology ORA, I mean ORA based geo analysis. I chose it, and I choose the algorithm, hypergeometric algorithm, and the correction method is ACE corrections. Then do the analysis. And it gives me this, like, it's a biological process category, and this is the molecular function category. And if I say cellular component category, but if I want to combine all, this is also okay. Then all, or better one by one, I can export the data or figure here from. This is kind of I can export the figure as a PDF or PNG file. Then one by one I can save in my directory, and I can also download this analyzed data in Excel file. I can also have a look in details. For example, cellular. If I want to have a more interest on one particular geo, I can go a little bit detail here also. This is kind of advanced check on. This is the additional things for a particular geo if you are interested in. Then if I go for a next source of analysis, for example, transcription factor binding site analysis, I can go the same way. And then shortly I can do this, and the TRA, and it is the default. I can go forward. Then it comes like this. This is only a single database, SysRed. So this sorts of, that means two transcription factor binding site, we get it significant here. And the same way, if we go for another source of analysis, network analysis, but for network analysis, it is not compatible. Actually, it is not at a single go stops. If we do the network analysis here in these tools, I must go for another tools to visualize this network. That's why for network analysis and interactor analysis, I usually choose a complete tools that I can do a network analysis and I can visualize my networks. For that, that's why I choose a network analyst. So until now, for this InetDB, this is, I think, is okay. It's a kind of demonstration. This is not to make you understand fully, just a kind of inspiration to go further later on. So if I go back to my previous slide, how can I go? I'm sorry.
You can exit from the share or exit from the software and then back to your PowerPoint. Okay. Or just back to your PowerPoint and then slideshow that one. Back to your PowerPoint. All right, okay, perfect.
This is okay?
Yes, yes, yes.
And I had a plan to show another tool that is for the network analysis. Here I use the data from Dr. Asif. It's about meat quality data from sheep. We recently analyzed this data. So let's go for this tools. This is network analyst. Oh, I have this another. So can you see my screen now?
Yes, it's clear.
Okay. Okay. No, not in PowerPoint. It's in another screen. I'm now. So I'm talking about a network analyst. It's an online open source tool, bioinformatic tools that provide a comprehensive analysis for gene expression data, meta-analysis, and network biology. And it's a recent development. I think it's 4 or 5 years back from a Canadian group. And I think I found it's very user-friendly, not so complex one like other open source tool. There are 4 options are there. One can go with a particular gene list. Here is gene list, multiple gene list. One can go directly from the data expressions, what we got after the normalizations, microarray, or normalizations of read count from RNA-seq, and also very preliminary raw data from RNA-seq we can go with. So I'm going to show the list of differentially expressed genes I have ready in my hand from RASEP. I'll go with this, and I'll pick the data from sorry. I'm taking data from Dr. Asep. It's about meat quality in sheep. I took the list of differentially expressed genes along with the fold change, how much they change after the condition. And because I changed that gene identifier to human orthologous, that's why I chose human species. Then I chose its official gene symbol. I uploaded the official gene symbol. Then I can click upload it here. Then I need to proceed. And it's an instruction window. What the way I should go, I need to go. I can follow a lot of options are there. The first 2 categories are accessory assorted visual analytics. This is kind of functional analysis related with gene ontology, pathway, and list of kind of heat map or Venn diagram or chord diagram. These are the things. And if I think about the network things, then second option is network visualization. And different types of networks are protein-protein interaction network, gene regulatory networks, disease, drug, and chemicals networks, gene co-expression networks. Say, for example, gene regulatory networks, the protein-protein interaction network, genetic, kind of generic or tissue-specific. We can go for both. Say first, generic one. We have our data, and we need to choose a database from here. I think string intercom is a very rich one. We can go with this, and we need to provide a threshold here. We can put 500 like, and then okay, we can go forward, and it gives us a potential list of networks. At least 8 networks we have, and this process usually the tools give us one big network, we call this continent, and some other smaller network, kind of independent, dependent with the bigger one. So if it is a very big number, then we need to sort it out to clear visualizations. I think it's only 676, then we can go forward. It's quite visual. If the number is so much, then we need to sort out, keep the number of genes in a short range. Still, it's a very big one. And then we should choose the topology, and we can choose the background is white. Then we can say, for example, fix or change the layout. It's auto layout. It depends on the user, and it's kind of subjective matter, and how he choose and how the outcome looks like. And then it's okay. When it looks good, then you can export the figure light. But until you can go, you can trial, and you can try, you can exercise with this, you can play with this. Once the best fitted or best attractive figure you can find, then you can just extract and change it, and different ways you can trial. Once you got this your choice or lucrative figure, then you can go with this figure extraction. The way is download. For example, PNG, you can export the PNG files, save as like this, and in your folder you can save. This is the way you can go for. And along with this, you can save this which we call this node table. All the informations are here, particularly two numerical values we consider to select the best potential gene. For example, network analysis we do with two purposes. One is that which genes are the key genes that regulate most of the genes of this network, and what the functions they coordinately control. So for this, we need to select, or we need to assess two parameters. We call this centrality parameter. One is degree, another is betweenness. Degree means how many genes interact with this particular genes or node. And betweenness is kind of interactions that not only this one-way interactions, it's go through and connect with another node or gene. These two parameters, based on these two parameters, we do a statistical test for significance parameters and weight-based weighting subsequent module detections. And this way we can choose this best 2, best 5, best 1 genes. We call this hub gene. That is most potential regulatory gene of this network. So there are lot of network exercise you can go for. I just show a protein-protein interactions networks here. And you see, and there are so many options are there to go for. You can choose other networks here, like gene regulatory network, transcription factor and gene interactions, transcription factor, microRNA interactions, protein and drug interaction network, protein chemical interaction network, gene disease network, and tissue specific co-expression network, and cell specific co-expression network. All goes the same way. You need to customize at your choice or desire. So I think this is all about a short demonstration. If some of you are interested in, just go through. There are tutorials are here. You can find a lot of tutorials, and if you just spend some quality time here, definitely you can learn the things very well. So with this, I hope you very best, and I'm going to close my talk. So in a nutshell, I can say for what the necessity of bioinformatics stops for biological interpretations of transcriptome data we have from RNA-seq or NGS or microarray. So whatever RNA-seq and microarray big data, after statistical analysis, we get a list of differentially expressed genes. That is the initial input for bioinformatic analysis. And nowadays, so many free open source bioinformatic tools are available. There are a lot of analytical options out there. You just need to spend some quality time to learn about. And it depends on how much bioinformatic you can give input in your data, and that much functional or biological output you can get from there. And for this, for the beginner, you should choose a very much user-friendly and one-stop as integrated tool that have the more visual power or more graphics power of these tools. You can choose for your initial trial or analysis. Then one by one, once you get expert, then you can choose other ones. So with these things, I would say thank you very much. If you have any questions, I'd be happy to answer you, please. Thank you.
Thank you, Dr. Aminul Islam, and also thank you for Dr. Ashraf also for the talk, and also for the very interesting demo, Doctor. Yes, we do have some questions waiting for two of you. I think now is open for Q&A sessions. I already received several questions from the committee, but I think due to the time shortage, I only pick some one or two questions for each of speaker. Maybe I would like to start with Dr. Aminul Islam.
Yes, please.
The first question is from Sigit from UNS Solo, National University Solo. All right, listening to your talk, remind the doctorand talk in the Husserl. So the question is, since there are many bioinformatic tools that are available only for human, what is the best strategy to overcome if I am going to perform the sequence in cattle? Do we need to align one by one? Okay, I think you may.
Yes, I got the questions and very intellectual questions. It is. I like these questions, and it's the question should come because it's obvious there's a lot of bioinformatic tools are available, and those are online available. I mean, freely available. And most of the guys or most of the computer people are focused to develop the software that is related to humans. You know, it's a sensitive case, and it's a very ultimately it's a money matters here, and that is the big things in behind. But yeah, still there is a hope. Particularly cattle and pigs and chicken are coming up with the bioinformatics and molecular genetics database increasing and improving. So for particular case, one person's dealing with cattle stuff, he or she first look for open source tools if already available and enriched with cattle specific, I mean cattle genome database, cattle genome informatics. And it's not a very simple questions or simple task to choose. Lot of trial and error he must do, and he need to check some sort of quality check or questions in mind, what he actually want to have from his data, what is the questions he has in his experimental design, and how much risk the cattle, particularly that lines of data. I would say, for example, cattle reproductions, cattle immunology nowadays is more richer than if I compare 5 years back. So day by day it's increasing, but still there is a questions whether it is sufficient enough or I need to shift human genome. It's up to you. If the persons or the biologist satisfied with the things or particular host he is interested in, if he didn't think that it's not sufficient or he can give a trial to compare to human genome, if the human genome output gives a better things, better logs, attractive output or figure, then he must go for the human after the conversion of the identifier. Otherwise, if he got a satisfactory output from the cattle specific genome information, he can display, because it's his pride. He's dealing with this particular stuff, and the analytical I mean algorithm behind or informatics behind is particular species specific. That is more convincing to the reviewer during publications. Reviewer always sometimes they just push back and ask why you analysis based on the human genome, whether pig or cattle you are working with. There is at least some sort of informatics resources are there. This kind of things later on he or she could face. So this also need to consider during this analysis. It's kind of exercise or kind of play, and at the point you satisfied, then you can go forward. Something like I would say.
Okay, Doctor. I think it's very clear. I hope the participants, Sigit from UNS, is clear enough with the answer. UNS, I have to correct the full name of UMS. It's not University, National University of Solo, Universitas 11 Maret, National University of 11 Maret, whatever. Okay, so all right, the second question goes to both speakers. I think Dr. Aminul maybe can answer first. From Asiana Hairunisa. Let's say if we have no reference sequence that are available in online database, can we still do the RNA sequencing study? Perhaps Dr. Aminul can answer first.
Sorry, just can you repeat me the question?
If we have no reference sequence available on online database, are we still be able to perform a sequencing, RNA sequencing data study?
Yes, still you can go for, but hopefully whatever the draft genomes ensembling is not perfect enough, but almost many species are coming. So I think major livestock species and human, of course, forget about human, many livestock species particularly are already growing, and some are particularly if you think about the reference genome, it's already established, ensemble established and getting updated time to time. So I think it's not a big worry about the reference genes particularly. Maybe it's not a very updated one, but still you can find the genome of your interest. And if the case is that if you don't really found your particular case for a rare species you are dealing with, then there's some sort of alternatives are there. Or the way I can speak. Yeah, that is a little bit complex, but still there is some sort of solutions for this particular complex case. Whether you can go for, an alignment with these genomes, you don't have a particular reference with this, but still they have. But what I would say is I hope that all sorts of genomes, particularly is draft or complete different ways already available, at least last 4 or 5 years.
Okay, Doctor. I will shift to Dr. Asep. Do you want to add anything? Because
yes, of course. Yeah, thank you for the question. Maybe my simple answer. From time to time, the reference genome is updated. Yeah, for example, when I use the SARS-CoV genome 2012, it is version 9.1. And then suddenly after 1 year, they already update. And then we use the currently reference genome, version 10. Yeah, so you can imagine in 1 year, and then they already make a repository to become more complete. So that's why our contribution, like when we perform RNA sequencing, normally from the reviewer, they ask about the repository, our sample. Yeah. So normally we deposit in NCBI, and then we get the GEO number or the ID number that belong to our experiment. So, and then in the next, when we repository, they will make an update for the reference sequence to become complete. Like I mentioned, when 2005 or 2006, maybe when we alignment, it could be only 50 or 60%. But nowadays, maybe like our previous study in 2019, we can align, we can map to the reference genome until 90% or 91%. I think that my question. So at least since livestock, maybe we can use reference sequence. But if not yet established, for example, when we work with buffalo, I don't know, buffalo, maybe some people still make orthology to the cattle, for the blast or similarity like that. Thank you.
Thank you, Dr. Asep and Dr. Aminul Islam. So means is without the reference sequence, so it's still available, still doable for the RNA sex analysis. Okay, last question for Dr. Asep. For Professor, I'm sorry, I need. The last question is from Professor Dr. Suyadi. Is it possible to isolate messenger RNA or total RNA for RNA-seq from blood sample?
Yeah. Okay, thank you, Professor Sudi. My greeting to Professor Sudi, Dean of University of Wijaya. Yeah, normally, actually, I'm not yet dealing with this liquid sample. Yeah, but maybe it is also possible since we characterize now sperm. Yeah, it is also liquid. We already establish our good RNA quality, but of course, it's not really so easy like from the tissue sample. We have to really optimize the protocol and to modify. Yeah. So to reach the RNA integrity, yeah, 7, it is quite tough. Yeah, but at least with some improvement, and then we get. So the thing also that from the liquid, we have to preserve the quality of RNA. So normally they suggest using ethanol, yeah, alcohol, to preserve the quality. If we are not preserve in the ethanol, it is maybe in one day it could be decreased. It could be degraded. Yeah. For example, we have experience, yeah, with this sperm. It is also liquid. And then when we running for the sequencing, yeah, only 1 or 2 days, it's become 1 or 2, only 2 RNA integrity. So it's more not. I mean the blood maybe, or the milk, or maybe the sperm, it's will more degenerative. So that's why we suggest to preserve, yeah, using ethanol like that. Yeah.
Oh, hi, Asad, can I jump in?
Yeah. Okay.
Yeah, please. Hey, it's not. You can easily isolate RNA from blood, but what you have to do, the majority of the cells, the most of the cells in blood is actually RBC, and we all know that RBC has no nucleus, so
yes,
no RNA. So what you have to do, just use histopack. It's a density gradient things. Use histopack, and you will be able to get rid of all the RBCs. You will get the Buffy code that contain all the cells, granular cells like WBC, then monocyte, lymphocyte, everything together packed. It's called Buffy code. Get this Buffy code, and then you will be able to isolate RNA. It's called histopack density gradient. You can do it. It's very simple technique. Thank you.
Okay, thank you. Because I'm not yet have experience actually with blood, but maybe Dr. Jasim has experience, and it's good to share with us. Okay, thank you, Dr. Jasim.
Oh, Dr. Asep and also Dr. Jasim to hop on. Welcome also to the webinar. All right. Okay. Thank you for both speaker. We have many questions are in queue still, but I think the time is very limited. So I would regretfully I would like to stop the first session now. Thank you very much for all the speakers and participants, participations. To the speakers, would you mind to hold on your video? I would like to share the token appreciations to both of you from the committee. Unfortunately, due to the pandemic, we cannot share the token appreciations directly. So the committee has already prepared a certificate for you. That will be also sent to your email after the event. So what I can do here is I just display here the certificate for this. The first certificate goes to Dr. Asep Gunawan. Thank you very much for being the speaker in this session, and then also for Associate Professor Dr. MD Aminul Islam, far away from Bangladesh, joining with our first sessions this morning with other speakers today. Thank you very much for both of you.
Thank you.
Thank you.
This will not the only collaborations between department and speakers. Also, we will have more real collaboration. Have a good day, and thank you very much.
Thank you.
I think I will proceed to the second session. I think the speaker all are here. We have Dr. Jasim Udin from also Bangladesh Agriculture University, and also is just one. And Professor Mehmet Ulas Sinar, are you here? Can you hear me, Prof?
Yes, I can hear. So can you hear me?
How are you? Hey, yeah.
Thank you very much.
All right, okay. So everyone is here. I think I will just go to the first speaker for the second session. The first speaker in the session will be Professor Mehmet Ulas Sinar. Before he starts his talk, let me brief you his CV. Okay, I hope everyone can see my screen. Can you see my screen?
Yes.
Yeah, so we can.
Okay. Okay, the second speaker from Department of the third speaker for today for today's sessions is from Department of Animal Science, Faculty of Agriculture, Erciyes University of Turkey. Do I pronounce correctly? Erciyes University of Turkey. Professor Mehmet Ulas Sinar is from Turkey, and then holding a position as associate professor in animal science at Erciyes University. Earned his bachelor degree on Faculty of Animal Science in Ege University Izmir in Turkey. Also, SAM University from his Master of Science in 2006, and earned his PhD in University of Bonn, Germany, SAM University with the previous speakers. He is very prolific researcher, publishing more than 1,600 papers with H-index and WOS is 17th. Google Scholar is 24, and then I-10 is more than 40, with total citation is more than 400. It's very good record. And more than, it's about 38 papers published in Q1 and Q2 journals. He also received several prestigious awards, including ASAG Travel Award in 2016, International Society of Animal Genetics, and also European Biotechnology Congress Best Oral Presentation Award in 2015, Chamber of Agriculture Engineers Turkey Early Career Award in 2014, and then another post award from ISAG in 2012, and also Travel Award from University of Bonn, Faculty of Agriculture. He has experience a lot in the international lecturing, agriculture, science, and technology graduate program in his home university, and then Erasmus lecturing in Ege University, Győr University, University of Bonn, and then Udin University and Debrecen University in Hungary. I think that's all for the CV is still long. I think it's enough time to read a whole CV. So I think I will just give the stage to Professor to start the talk. Prof,
yes.
To start the talk, I hope you can finish your talk in 30 minutes.
Yeah, thank you.
Just let me know if you need extra time. Thank you.
Thank you very much, Mr. Modoy. So it's early in the morning, so I would like to say good morning from Turkey, actually. So thank you very much again. So, but I need to make, I think, a screen share area. So how to do it? Let me check. So share the screen. So can you see my presentation? So I think, yes, this one. Share.
All right, okay. All right, go ahead.
Can you see it?
Yes, we can see.
Okay.
You can just start with slideshow maybe. Is it
Yeah.
slideshow?
36 slides here, so I have.
All right, okay.
So thank you very much. So good day to all. So my name is, as Mr. Budiman is introduced, Mehmetullah Çınar. So I'm working RGS University as a full-time professor, and also I'm working at Department of Veterinary Microbiology and Pathology. So in the College of Veterinary Medicine at Washington State University as an adjunct professor. So today I would like to speak about the basic principle of real-time PCR validation for holistic RNA expression. The holistic RNA expression, the bioinformatic tools for RNA expression actually. So for mainly holistic RNA expression to up-to-date RNA expression, because there are some older methods for holistic RNA expression also. So it's mentioned about Dr. Aminul Islam. Then I would like to mention about this session, so how we can validate the results of the RNA-seq experiments, how we can validate the genes coming from this as an outcome of these experiments. So as you know, the holistic gene expression is mainly done with the two methods. So the first method is, the current method or the most up-to-date method is RNA sequencing. Is done by the different platforms actually. So yeah, the most famous, one of the most famous actually platform is the Illumina, and more newest is the Pacific Bioscience. And so I don't know how to apply, but I never worked with this one. But in the literature also it's mentioned that Oxford's Nanoporia. So all these methods are RNA sequencing. They are sequencing all platforms in sequencing the RNA actually, and by different using chemistry or using electronic techniques or computer techniques, but all based on the sequencing of RNA in this sophisticated instruments. But the outcome is coming after the sequencing. It's just simple, like a Sanger sequencing, base by base reading actually. And when we annotate this base by base reading with the like Ensemble or NCBI, so the organism's genome annotation tools or bioinformatic tools, or so when we annotate them with the computer softwares, sophisticated softwares, we outcome with the genes. And the other method is more, how can I say, not very up to date, but it's still used. So it's the gene chips. So it's just based on when you put RNA on a glass slide, so the RNA molecules are hybridizing with the artificial oligonucleotides on the glass slides. And then so the laser scanners are reading this annotations, or we can say the hybridizations. Then the computer software is changing or alternating it to the genome information or gene information. But why we are doing these all approaches actually? So by RNA sequencing or microarray experiments, or so whatever, maybe there are some coming new tools. So for measuring the expression or measuring the genome's transcriptome actually. So the main reason is, since we would like to understand the genetics, but genetics is not only the genetics. Genetics is consisting of different levels, we can say that we call them system genetics. But system genetics is normally shown is like arrow from top to bottom, but it's not like that. So they sometimes, they started like central dogma. It's just not moving from top to bottom. So it's sometimes interacting with each other. So then we can show it more. Actually, I found this one more precisely or more correct. We can show it as triangle. But understanding the part of genetics is also consisting of transcriptomics, and transcriptomics is not only transcriptomics, it's consisting of epigenetics or expression QTL, or to understand the transcriptome variants, how it is associating with the phenotypes actually. All understanding those, how can I say, complicated relations. So we need to validate we need to measure first the gene expression by using gene chip or RNA sequencing, but then we should validate the genes, what we are handling from this kind of experiments actually. So nowadays, you can see not only RNA is not only RNA is also consisting of many different RNAs, And so the companies, so the commercial companies are providing RNA sequencing actually. So for example, you can read the RNA by single end sequencing, means that single direction read, or so you can make a double direction read. Even you can nowadays, so we can harvest the expression of the microRNAs or small non-coding RNAs. These are so indifferent. For example, the prices are day by day decreasing, means that it is getting easy to access day by day to the RNA sequencing or transcriptome analysis. For sure, it will be more cheaper until a new method comes. So, but as I said, so always the transcriptome measuring methods or understanding the transcriptome methods is changing and so developing actually. Most probably in coming days, maybe new tools or new computers or new systems are going to be launched. So, but the prices are, yeah, so maybe in the beginning is the high, but then it's decreasing.
Sorry,
Prof. Ulas, can you share full screen?
Yes, sure. So I don't know how to do.
It's not in full screen. Maybe you can.
So let me.
There is an option whereby you can hide the presenter view. Somewhere on the icon below your screen.
Select QSTC. This is the new version of. So that's why.
Somewhere beside the what is it? The search icon, Prof. Below your slide.
Below my slide, so I'm doing. So this is not, I think.
There is a icon of the half screen, I think.
Let me. So this you can see. It's in the slideshow at the moment, but you cannot see in
I know. It is in the slide screen, but what we see is what I call it. It's only half of it. Oh yeah, that one. Can you click that one? No. How about the three dots over there? The three dots. Can you click three dots?
Yes, yes.
Yes, and then I don't know. Is it hide the presenter view?
Hide the presenter view. Ah, yes. Maybe yes. This one. Yeah. Now it's okay. Yeah.
But we cannot see. Can you?
Yeah.
Can you turn off your sharing and then back to share the?
So can you see at the moment?
Can you slide show now? Can you try?
So I don't see it. Yeah. Is it okay now?
Perfect. Yes.
Okay. Okay. Can you? So means that. So sorry for inconvenience. Actually, I'm sorry. So I'm not that much good in the.
No worry.
New this Windows tools, so it's always complicated, and they are changing the features of this kind of stuff. Yeah. So qPCR. Sorry again. So qPCR is widely known as the most effective method. So to analyze as a qPCR means is actually known, so maybe it's an abbreviation, but it's more normally about the real-time PCR. We know more about the real-time PCR. So we can abbreviate real-time PCR as a qPCR, quantitative PCR. So qPCR is widely known as the most effective method to analyze the modulations of the gene expression. Why? Because of its efficiency to detect and precisely quantifying the target genes even at low expression levels. Means that this method or these instruments can understand the expression of genes even if they are very low. So these instruments or this system can identify the expression of what how can I say, gene chip or RNA-seq cannot catch. This system can catch actually. So what is the Dr. Aminul Islam is mentioned in the conclusion? So differentially expressed genes. This is the term which we are hearing so much actually, that if we are doing the transcriptome analysis, we use or we hear this term so much, differentially expressed genes. So what is the differentially expressed genes? A gene is declared differentially expressed if an observed difference or change in read counts between two experimental conditions is statistically significant. So to give a more concrete example for that one, so I think most of the participants are animal scientists or from the agricultural. For example, if we have if we have a slow growing animal or high growing or fast growing animal. So if we can get the liver of those animals, if we compare the transcriptome of the liver from fast growing and slow growing, which genes are differentially regulated between fast and slow, means that these genes are affecting in the genetics of the growing actually. So these are the differentially expressed genes, and there are different statistical methods for differentially expressed genes. So for example, there are parametric methods. So if it's binomial Poisson, negative binomial distribution, or non-parametric, these are the, how can I say, different tools or methods or adds in for example, our program. So these are the statistical framework that maybe the Dr. Aminul Islam I missed, but sorry. But he mentioned about these statistical approaches more detailed in his presentation. But what we are getting after a RNA-seq or microarray experiment. So we are mostly doing. So this is an example paper. So from the Nordic dairy cattle, which is done for high and low residual feed intake. So this one of the left. This is the left side. So we come up with these heat maps actually. So this is the most famous, one of the most famous. So a visual, how can I say, graphic methods for describing the RNA-seq results. And one of the, this is, I think, the purple part is, for example, representing the high feed intake, and the green part is representing the low feed intake. And as you can see, the different colors between two groups is representing the two, how can I say, two different, for example, gene expression. And these are the genes. So which genes are differentially regulating between, for example, between the two groups. And these genes are come out from the RNA-seq experiment. But the question is, are these genes are really correct? After the RNA-seq or mainly from the microarray expression, is this gene really, for example, just assume that FDXR, is this gene is really differentially regulating between high and low residual feed intake? And how we can, the question is how we can validate it. So is this the really down-regulating or up-regulating genes or differentially regulating genes between these two groups? And so we are using real-time or qPCR analysis to validate, to confirm the results coming from the actually RNA-seq experiments or microarray experiments. This is just a pipeline of regular RNA-seq experiment. Is coming from the raw RNA-seq data, and we align. For example, we align with a genome annotation. For example, if we work with the chicken, we align to the chicken. If we work with cattle, we align to the for example to the cattle genome. Now, as Dr. Asep mentioned, the genomes of livestock is very developed, just exceptional with the some organisms, like he mentioned buffalo, for example. But I think in very near future, the genome of this kind of organisms is going to be completed, and then we will be able to align this also sequencings to the it's the buffaloes itself actually. And then the gene quantification after we aligning this sequencing results to the genome after we make the genome annotation, we make the gene quantification, means that we understand the quantifying how much the genes are differentially regulating between for two extreme cases, for example, like the previous example was high residual feed intake and low residual feed intake, and then normalization as we did in the regular RNA-seq experiments, and then we identified the differentially expressed genes analysis. And after we doing differentially expressed genes analysis, like shown here, we need to validate the genes which is popping up after the bioinformatic tools. We should validate them actually. But not only the qPCR, or we can say that real-time PCR, not only used for gene expression validation studies. There are several other actually aims to do that one. For example, nowadays it is very famous, or it's very hot topic in the world. Genetically modified organism analysis. We are using real-time PCR for this issue also. We try to understand, or we should try to differentiate. The commercial, that means that not commercial, the conventional plant or animals from the genetically modified organisms as well. So it could be also microorganism. So genetically modified microorganism, animal or plants, how it is differing from the conventional ones. Yeah, so, and then, so we are using the qPCR or real-time PCR for sample verification. So it means that whether the meat, for example, sometimes it is happening also in commercial food industry. So they claim that it is used for cattle meat, but the cattle meat, it is not used. So for example, to identify the amount of, for example, chicken meat inside the cattle products. So we can differentiate the amount by using qPCR also, or plant pathologist, maybe if there is a plant scientist inside. So we can understand the load of the pathogens, viral, bacterial, or fungal, how can I say, pathogens on the plants. We can detect these ones with the qPCR analysis. And there is also one less used, maybe pasto-authenticity. So it means that the wheat genetics is mainly also so identified. I think these food scientists mainly use the sample verification instead of animal scientists. And also another famous, as I show, so nowadays we are not only using transcriptome with messenger RNA or total RNA, we are using the transcriptome, the microRNA expression profiling is also very famous. But so the microRNA expression profiling is quite different than the normal messenger RNA expression profiling. It still requires sophisticated, I mean that, primer system is just, how can I say, provided by companies, with custom, as a custom, how can I say, designed primers, so that we cannot design these primers on our own computers by using some free programs. These are just if we assign the sequence, the companies are providing these primers with the labeled probes, sophisticated probes, they are delivering ready use actually to your laboratory. But we can still using the real-time PCR, also validating the transcriptome coming from the small RNA sequencing as well. So to apply real-time PCR or to quantify or to validate the gene, first you need a real-time PCR instruments. Nowadays on the market, you can find a lot of real-time PCRs. They are also developing day by day, but some of them is a little bit more famous or more consumed or more used, more applied for in the labs than others. For example, one of them is the Roche Light Cycler 480. It's 96 well. Very famous, real-time PCR instrument. And then the Applied Biosystem, this is quite the old one, 7500, but nowadays Step 1 Plus, for example, mostly the commonly used one of two methods. And for example, there are other smaller scale instruments also, Bio-Rad or other companies, all they have different kind of instruments, which are used for real-time experiments. But the idea is always the same. When the machines or the detection methods or inside instrument components are different, but the idea is always the same. This is the left one is the right one is quite, it's the old, how can I say, method, or old technology. There was a tungsten lamp for as a light, how can I say, source, but nowadays as a light source, the LED light sources are used in the current instruments, but the method is very, very simple. There is a normal thermal cycler is inside the machine, actually means that the machine can adjust the plate or I mean that the cocktail, how can I say that, cocktail temperature as a regular thermal cycler. But there are some camera or light sources, which can, how can I say, which can identify the emission of the light. Then when, for example, a primers or whatever, is hybridizing with the DNA or RNA molecule inside the machine. Then a hybridization is occurring, and the chemistry is allowing, when there is a hybridization occurring, the chemistry is allowing a light emission, and the machine connected to the computer, and the computer is converting these light emissions to the signals, and then allowing scientists to understand the quantifying or understand the or observing the DNA amplification online. That's why, so it's called real time, means that, so when the DNA is amplifying on the plate or wherever you use on the tubes, or so as a cocktail, chemical cocktail, when it is amplifying, then you can, other than conventional PCR machines, you can observe the amplification real time. That's why these machines are called real-time PCRs. So this is other than conventional PCRs. The idea is the same, but when computer programs can turn these signals, so on the thermal cycler, so then it's called real-time PCR. So to make a real-time PCR, there are some steps that I will introduce you. So now, so it's not that the step one is as I said, the PCR step. So it's not that much different than the normal PCR step. So there is a denaturation, so then annealing, and then the extension. It means that these are the steps that, so the DNA is amplifying actually on the tubes or on the plates here. And so the most important, one of the most important actually the cases for or steps for RNA sequence or RNA validation, so qPCR experiments, is the sample purification or quality control. So when we are actually doing transcriptome or qPCR or whatever the transcriptome or gene expression methods, we are starting with the total RNA isolation. Actually, the total RNA has historically been used to describe extracts containing the traditionally defined RNA types. These RNA types are many, actually, not only one. So so there are ribosomal RNAs inside, transfer RNAs inside, so small RNAs, so large non-coding RNAs inside. There are maybe 30 types of different RNAs in the total RNA. But so since we would like to measure the gene expression, so we are targeting actually, so the messenger RNAs. So for validation of the results, unless we are using microRNAs actually. So if we are using microRNAs, our target will be the small RNA portion of the total RNA. But if we assume that we would like to validate or quantify the messenger RNA, we are mentioning, so, or we are focusing on the messenger RNA inside the total RNA. But unfortunately, so the total RNA is huge, but the messenger RNA is very low. So means that only 2 to 5% of the total RNA is messenger RNA. And there is always chance to miss this messenger RNA when you are isolating the total RNA actually. So this is the reason that when you make an experiment and when you don't see any results at the end, so maybe you will be able to, or you can be, how can I say, not to isolate the messenger RNAs from the total RNA. This could be the one reason. Or for example, purification of smaller microRNAs other than non-coding RNA molecule offer additional challenges due to smaller size. But now thanks to chemistry actually, nowadays there are very nice or sensitive even messenger RNA isolation or small RNA isolation kits. Very sensitively they can isolate the small RNAs or messenger RNAs, but there are always, so especially for new users or beginners, I mean that they can make these mistakes, although they have the sophisticated kits or chemicals, so they can miss these small RNAs or messenger RNAs due to their, how can I say, amount in the total RNA. But so we can measure, how can I say, so in the middle spectrophotometry based systems, we can measure, for example, the quantifying the amount of the qPCR. So we can measure the amount of the total RNA actually by using spectrophotometrical techniques like nanodrop or other tools. So for example, the spectrophotometry-based systems are the most famous ones. So it's the most of you maybe knows about the nanodrop. So it's measuring the light emission also. So the wavelength of the items inside the liquid, for example, it's measuring in 260 nanometer and 280 nanometer, and the ratio of this 260 nanometer and 280 nanometer is important. So for a quality or for a good gene expression quantification, so it should be 2 actually. But this 2 is not always so constant. it can be vary due to even so resuspension solution, the water even, for example, if you use if you use not appropriate water for resuspension of the RNAs, when you after you isolate the total RNAs, if the pH of water is different, for example, or if you are not using the appropriate water, highly pure water, for example, so the pH of water or other minerals inside the water can change the RNA content. So for example, as you can see here, so due to types of the water, the RNA quality can change from 1.5. The ratio of A260 and A280 can vary from 1.5 to 2. Means that even the water is important. You should think about the water quality, what you are using for total RNA isolation, resuspension, the RNA to keep in the refrigerators or freezers. So these are all we can quantify, or we can check the quality of total RNA by using fluorescence-based quality assessments. So, but these are more expensive methods. If you have a very sensitive RNA, or if you have very sensitive experiment, so that you are working with maybe picomolecules, so maybe you need to, how can I say, quantify your RNA by using this kind of fluorescence-based quality assessment systems, but they are not that much, how can I say, cheap experiments. And some of them we know them, so very famously ethidium bromide, for example, or cybergreen. So these are the most common used actually fluorescence for how can I say, measuring the quality of the RNA. And also the other problem for qPCR analysis, contaminants and inhibition actually. So for example, the main contamination or most possible contamination of RNA is with the genomic DNA. So we should be very careful on that one. So if you, for example, if there is a genomic DNA, so is mixed inside the total RNA, then the results will be, how can I say, biased. So the results will not be the true results. It should be, how can I say, false positives actually. So that's why we should always be aware of the genomic DNA contamination. So we should maybe use our DNases so to digest the DNA from the RNA experiments. We shouldn't forget that one. Or the inhibition. If the test sample contains an inhibitor, such as water, for example, even water could be a inhibitor. So this is also affecting the qPCR results. And also, so the template degradation. So we can check the template degradation with Agilent bioanalyzer. This is, as I don't know, but this is still only the tool that to measure the template degradation. These days, it was 10 years before only the Agilent bioanalyzer, but still it is used, I think. Or so we should use appropriate negative or no template controls. So these are important to understand whether our RNA is correctly isolated, or is there any messenger RNA inside the samples. We should use this negative or no template controls, or to check our water what we are using. This is, or whether is there any genomic contamination inside the water what we are using, or inside the genomic DNA contamination inside the messenger RNA. We should check by all using this positive or negative controls. Or reverse transcription priming, two-step RT reactions. These are always important also. So this reverse transcription means that converting the RNA to the DNA. So this is also an important and sensitive step that can affect also the quality of qPCR. So if you miss to put one chemical, so then you can how can I say, face with the no result or with the biased results actually. So it is generally assumed that all the RNA and messenger RNA in a RT reaction is converted to the cDNA, means that we assume that one, means that 100% the RNA is converted to the cDNA after we did the reaction, but this is incorrect actually. Not always the 100% is converting. So it's always some part is missing, or some parts, maybe it could be sometimes by chance 100% is converting to the cDNA, but not always occurring actually. And these are always the factors that can be, how can I say, affecting the qPCR. And another important step is the qPCR assay design. So means that replicates and randomization. This is always the question that are we going to test biological replication or are we going to test technical replication? Actually, this is upon your, how can I say, experiments. So, but just I would like to show, firstly, actually, I would like to say, statistically or what is suggested in the literature. So if you would like to quantify a gene, so at least you should, how can I say, make a 6 replicates. This is the, how can I say, the ideal amount. So for example, you may have different limitations. You don't have, for example, enough number of animals, or if you don't have, for example, enough number of samples, or maybe sometimes our samples are degraded. So when we are making the experiments, actually, so we may miss some samples. So this is the ideal case that 6 replicates should be done. So for a successful qPCR validation, but this is not must, actually. This is the ideal. But so I would like to mention about the biological replication and technical replication. Biological replication is representing actually the different subjects. For example, different patients or different animals. So the sources coming from different patients or different plants or different animals, these are all representing the biological replication. So, and it is going always parallelly, actually. When you started with subject or animal or plant 1, so you should go all through with the subject 1, animal 1, plant 1. But so if you have a technical replication, so if you want to make, for example, validate your, how can I say, samples, it's not only, so you don't want to understand the biological variation, you would like to understand your technical variation, you should start with the same sample, but then you can, how can I say, make from the same, how can I say, sample or animal or human, whatever, so you are using as a patient, for example, so you can extract 2 times. So to check your extraction, so you can divide your samples into 2. So by the extraction, and then from this extraction, you can follow up with the different qPCRs, means that you can make run 1, run 2. So from the same sample, and you can compare your technical, how can I say, specifications, how I perform, for example, the isolation, how I perform the RT-PCR, so means that reverse transcription PCR, how I did the real-time PCR. So you can check about this kind of stuff with the technical replication, but this is not representing the different biological, how can I say, approaches. So means that this is upon scientists. So this is upon, how can I say, the aim. Yeah. So, but always there are 2 different, how can I say, approaches. Whether I am using biological replication or whether I am using technical replication. So primer, probe, amplicon. This is always, this is my also personal, how can I say, experience. So I did many real-time PCRs. That's why I feel that this is the primer design is very important step for qPCR, how can I say, validation. So always, so it says that, so for example, the unexperienced researchers or young scientists, so when they don't know, so when they're designing a real-time PCR primers, so whatever the, for example, the computer software is giving them, so they are accepting, and then so they are facing with, how can I say, unwanted results, actually. So since in the qPCR experiments, since our aim is not the sequencing of the, how can I say, the amplicon, so then we may always keep in the, how can I say, short sequences. And then, so the suggested one in the literature is always 75 to 150 base amplicon for primer sequences, actually. So, but the ideal one is around 100 bases. And the primer sequences should be 20 to 24 nucleotides in length for both forward and reverse primers, actually. And there are, for example, free tools. So to test the, how can I say, folding of the primers by using this kind of, how can I say, most probably these presentations will be shared with you maybe. So you can try these tools. These are the free tools, actually. You can, how can I say, you can check the folding ability of the primers which you design, actually. So, and then, avoid stem loop secondary structures with low delta G values, for example. So make sure that the amplicon is within a structurally stable section. So, and try to avoid palindromic sequences and try to avoid the maximal number of GC level. Actually, this is the 50%. Don't go for example, 55 or 60% of GCs. Always try to reject or try to make your settings, for example, always the primer designing tools is allowing, so what kind of GC content you want. So you can limitate, how can I say, the GC content. Try to keep, so these adjustments in the softwares, and avoid repetitive regions. For example, in the genome, some regions are so AT regions, GC regions, they can be repetitive, for example, 100 times, or for example, more, even 1,000 times. So try to avoid from this kind of sequences, not design your primers from that regions. So, and also avoid regions with SMPs in the beginning. So it was not easy to check these SMP regions actually. So, but nowadays the bioinformatic tools, or so for example, Ensemble or NCBI is very developed. That's why, so if you want to design a primer, then you can see even there is a whether any SMP that region or neighboring SMPs you can observe. So sometimes there is no, so we cannot avoid actually. So but so at least you can check. Nowadays the tools are allowing us to check this. So neighboring or SMPs even, or maybe if there is a SMPs inside the primer, for example, because if there is many SMP inside the primer, it's just, although it is just 20 base pair, maybe 2 or 3 sometimes SMP is, so how can I say, overlapping with the primer which we design, and then the annealing problems will come. So the primer cannot recognize the target part what you would like to amplify. Instead of this target part, it may, how can I say, amplify something else in the genome, which you don't want, and this makes also a false positive results. So you will see a qPCR result, but this is not your result actually. So that's why always to check the SMPs inside your primers. So that's always a good idea actually. And for transcript specific designs, so try to avoid design primers from intron exon boundaries actually. This is also, and nowadays this is also highly possible that we can very clearly see the exon intron boundaries inside the genes actually. So in human, rat or mouse, this is much more, how can I say, available, but always even in the how can I say, livestock or plants, nowadays we can see this intron exon boundaries actually. So if we design a primer in the intron exon boundary, so some part in the primer or amplicon can stay in the exon, and some part can stay in the intron, and then when you make a qPCR, this will be also a false positive results. So this is
Hello.
Yes.
We have less than 5 minutes left for your talk, Prof. Sorry.
Yeah, so sorry about, so I would like to go more so quickly. So there are, we can see that there are a lot of, how can I say, dyes for detecting the qPCR. The most famous ones is the Cybergreen actually. So now, the other companies also, so or other commercial, these are the registered ones, Scorpions, High Beacons. I don't know which companies, but so these are available. So also using as a light emission. So these are the fluorescence chemistry. So it means that, so the primer concentration optimization after you get the primer is also very important. As you can see in this graph is actually, so different primer concentrations resulting with the different qPCR results. So it means that these lines are representing the results of the qPCR. We can say that reaching the pivotal phases, and always so it is detecting with the different, how can I say, CT values. That's why you can see if you change your concentration of primer, you will face with the different qPCR results. And another point for primers actually the shoulder peaks. If this is the MELT curve analysis, so each so qPCR instrument is giving these results. So at the end, for your check, actually, if you see any kind of shoulder peak to your main peak, this means that there could be a primer dimer. That's why it's not a good. So then you can be, how can I say, think about your primers when you see something, right, results like that. And then another approach, so another step is quantification strategies. Then when you make a qPCR, then you will be able to make a quantification. And the number one quantification strategy is the standard curves quantification. And standard curve quantification is actually making a serial dilution with known number of copy of DNA inside your cocktail you can prepare, for example, from your DNA. So by making a cloning or transformation, so you can make 10 to 5, for example, from 10,000 copies to 1 copies or even or 10 copies. So you can make a serial dilution like that. And when you load this one to the inside, instrument, the machine can detect. So for example, how much the CT value is coming from the 10, how can I say, 10 copy, and from 10 copy to the 10,000 copy actually. And these are the CT values, means that first amplification of this is the number, cycle number, or CT value. We can say that this is the amount of, the DNA amplification signal is detected by the machine. And there are different, how can I say, algorithms, so in the literature to make the quantification strategies. This is a standard curve quantification, and there is also a second strategy. It's called delta CT or comparative quantification method. So it is just there is other than standard curve quantification. This time there is no standard curve. This is just you compare the cycle numbers you get from the machine, from your samples, and then you put this one into the algorithm that the ratio is given as actually qPCR results or gene expression validation. This ratio we are using to publish or for statistical analysis. And then another important step is the normalization. Since the RNA content means that the factors from RNA and from factors from the machine is affecting your qPCR results. For example, sampling, storage, quality, extraction, storage, yield, quality inhibitors, even the qPCR machine. Variation. To remove this kind of variation, you should normalize your, how can I say, gene expression with a normalizer. So selection of normalizer is a very important issue in the qPCR steps. Actually, this is a milestone paper. I suggest you to all read this paper. Actually, accurate normalization of real-time quantitative PCR data with geometric averaging of multiple internal control genes, always up to this. So Van de Soppelens paper in 2002, each of scientists is using only one normalizer, the most famous one even, the GEP-TIHA as a normalizer. So GEP-TIHA is essential cell biology or cell, how can I say, GEP-TIHA gene is very essential for the cells. So then we assume that GEP-TIHA gene found in every cell of the, how can I say, one organism. This is used as a general normalizer. But then in 2002, Van de Soppelens is suggested that no, one normalizer is not enough for accurate normalization. You should use 3 normalizer, and you should not use the you should not use the arithmetic mean. You should use the geometric mean of these 3 normalizer for a accurate normalization of your qPCR data. Then is coming that which genes is used for, which 3 genes or more genes is used for accurate normalization. Selection and validation of the gene reference genes. This is a paper recently, maybe 2 years ago, it's paper. This is describing, this graph is describing, this table is describing the mostly famous used normalizer or reference genes used in this kind of qPCR experiments in nowadays globally. Globally, all people, all scientists are using those ones. But which one of 3 is used to normalize our samples actually? To do that one, we can, how can I say, we can test our how can I say, which 3 gene is best normalizer among these so many genes actually. And to test that one, so the scientists are developing different algorithms as a softwares. For example, one of the most famous one is Genome, Norm Finder, Best Keeper, or we didn't use, but so nowadays, so Delta CQ method is also used for understanding the most stable reference genes in your biological material. And we also, as a lab with my colleagues, we tested, for example, several housekeeping genes or reference genes in different conditions. For example, we used how it is changing, how the stability of genes change with the age, or how it is changed with the LPS, lipopolysaccharide or lipoic acid challenge stimulation, or in which cell type. For example, this is different porcine tissues, this is different, so this is alveolar macrophages, or this is PBMCs. And all these three different conditions resulted with 3 most stable different genes actually. And this is showing us that these three combination should be set up by you, according to your cell type or your condition. This could be not easy for your cases, but it could be time consuming to do that one. But it's always suggested to handle how can I say, accurate normalization. And this is the Best Keepers website. You can read a lot of things from here. And nowadays there are some new-ish, this is really recently published paper. So as we said, there are 3 genes should be used for, how can I say, 3 genes should be used for accurate normalization. But in 10 years before or 5 years before, we were doing this 3 gene expression one by one actually. So we were measuring after and after that one. So gene 1, gene 2, gene 3. But nowadays, more accurate normalization. So there are some, how can I say, more reliable tools is developed, like multiplex, means that expression of measuring the expression of these genes in a same reaction. So to reduce the bias or reduce the variability coming by each running of the machine, so you can measure these 3 genes expression in one reaction. It is called multiplex. Multiplex expressions. As you can see, for example, all these 3 genes run in the one run actually. So in same reaction. So this is called multiplexing. And there is one paper, so maybe, I don't know, did I give the reference? So it's coming the end. So there is one, there is top of the table is giving. So this is a pipeline or check. It is something like a check sheet for qPCR experiments. You can see it seems to be easy for qPCR check always. But you can see there are a lot of steps to check actually. So then I suggest all scientists, researchers, to at least take a look on that one and try to apply or try to follow at least some of the, how can I say, checklist points, so to handle more accurate qPCR results. And so I use actually several literature when I prepared this presentation. But so this is very good, how can I say, manual, good practice guide for the application of quantitative PCR. So this is the first edition, but it's very useful edition. So maybe it will be, how can I say, updated in coming days. So I suggest all researchers to take a look on this manual for a accurate qPCR analysis. And it's the foreword written by the Michel Pfaffel. He is also developer of the Best Keeper program. He is very good in qPCR normalization or qPCR analysis. I would like to thank Dr. Asep Gunawan giving me this opportunity actually. And so I would like to thank IPB University Management for welcome sessions and technical aspects and introductions also. And I would like to thank also my colleagues, former colleagues, and still we are collaborating actually, Dr. Mohammed Jasimuddin from Australia, Queensland University, and so Dr. Aminul Islam from Tohoku University and Bangladesh Agricultural University. And I would like to welcome you to the Cappadocia, very nice volcanic region with fairy chimneys and special landscape, fairy chimneys and cave hotels with balloon, how can I say, air balloon, So flying, I can say that all welcoming and thanks for your attention for listening me actually.
Very much for a very interesting talk and also very interesting picture of Cappadocia. Cappadocia. Hopefully the webinar team will visit the sites soon in the future.
Yeah, welcome.
I think many participants impatient to ask the questions, but I think before we come with the Q&A session, I would like to call to the stage for the second speaker, Dr. Jasim Udin. But before he start the talk, let me introduce him with his brief CV. Let me share the file. Oh, okay. Sorry, Prof. Can you turn off your sharing menu? Okay. Sorry. I cannot share my files now.
Can you see now?
Yeah. Okay. Just a moment, Doctor. I will read your CV first for the participants.
Okay.
Can you stop your sharing screen first? Because I want to share my file first.
Okay, let me do that.
Okay. All right. Okay. Good. All right. Can everyone see my slide?
Not yet.
Not yet. All right. I think I already shared.
I still see maybe this Dr. Jasim screen.
This is
Queensland government.
Dr. Jasim screen. Dr. Jasim, can you? All right.
Yeah,
Okay.
I'm trying to do
Yeah,
that.
yeah. Now it's
All right.
okay.
Perfect. All right. The second speaker is from Department of Medicine, Faculty of Veterinary Science, Bangladesh Agriculture University. Same with the second speakers. Muhammad Jasimuddin, DFM, MS, PhD. He's now served as Associate Professor, Faculty of Veterinary Science, Bangladesh Agriculture University. He earned his bachelor degree from Doctor of Veterinary Medicine, DVM, from Bangladesh Agriculture University, as well as his Master of Science in 2006 from the same faculty in Bangladesh Agriculture University, and moved to German, the same university with the other speakers, University of Bonn, Germany. So it seems like today webinar is the reunion of the alumni of University Bonn here. Okay. Dr. Jasimuddin has published numerous papers in many journals with Edge Index of 20 in Google Scholar. At ResearchGate score more than 32, with number of articles is 68, and the total conference abstract of 58. So very productive researchers, and received several awards, including Global Research Impact Award from at the third level and first level at Bangladesh Agriculture University, and then selections 2012 was selected for the position postdoctoral researcher, Integrated Pig Genetic and Genomic Institute under Prof. Christopher K. Tuggle in Iowa State University. And then in 2011 and 2010, he received travel grants for attending the international conference in Greece and France, respectively, and also receiving a medal in 2006, a gold medal for distinguished result in MS in veterinary medicine and Bangladesh Agriculture University and the other awards. Okay, he has activity in teachings involving in the Faculty of Veterinary Science in his home university, and also in the Institute of Animal Science in the University of Bonn, and also in the University, Queensland, Australia, particularly in the School of Veterinary Science. So, Doctor, by this, I will transfer the step to you for starting your presentations, perhaps within 30 minutes, Doctor. Time is yours.
Try my best.
Okay, I will stop my sharing. Okay. Okay. Can you share?
Yeah, can you? Okay, can you see my screen?
Yes, yes, yes.
Everything is fine?
Yeah, it's still in your desktop. We haven't seen your PowerPoint.
Oh, really?
Yeah.
What is the problem then?
Are you connected to another monitor?
Yes. Is it? Can you see now?
No, it's still. Or is it? I already stopped my sharing screen.
Oh. Hmm.
If you are using the second screen, you can drag your PowerPoint to the main screen, maybe.
Yeah, that's what I have done.
Okay. I don't know what happens.
All the technical problem. Can you see now?
No, still cannot. Or okay. Maybe is it because I haven't leave my screen sharing?
Okay, yeah, you can leave it.
Yeah, but I already leave. Just one.
Can you see my screen now?
Still on your desktop.
Okay.
Doctor, can I reshare my screen first and then I log off properly?
Okay.
Yes.
Mm-hmm.
All right. Yes. Okay, share. And then I will stop here. Okay, I think I stopped my screen sharing properly. Now you can try yours.
No, is it okay? Can you see now?
Oh, still on your desktop. Alternatively, how to do?
Okay. Let me check now. So.
Okay, now it's okay. Yes.
Now it's okay?
Yes, then you can just start the slide. So.
Is it? Can you see?
Yes.
Yes, now it's clear.
Okay, please pause it, Doctor.
Just give me a moment. Is it fine? Can you see everything?
Can see. Yes. It's clear.
Okay. Sorry for this technical interruption. Thank you very much, the moderator, for a nice introduction. I'm actually working in Bangladesh Agriculture University, but since long time, I'm from Bangladesh Agriculture University as I work in Germany and now in Australia. However, it is always risky to talk at last because most of your brain has been used up by Asif, Aminul, and Olas. However, I will try to make it interactive and I will use very simple language. I'm promising you that I will not use any technical term. You have heard lots about the technical term this morning, afternoon. So everything will be very simple and easy. But what I want to tell you that this will be the very important session. because what you are doing, all the experiment fast and then analyze it. And end of the day, you need to publish those data. I will start with a nice story that happened to my life. When I moved to Germany, my PhD supervisor, what he told me that, look, there are lots of theses behind me, but nobody bothered about this. So I want the publication. And that's the first time I understand the importance of publication. Publication is actually representing you. Publication is representing your work. Publication is representing your lab, your group. So what is publication? I know that you all know about the publication. Publication is anything that we are publishing, but we are not talking about other publication. We will talk about journal publication, research article publication. It could be like research publication, could be popular article, like in newspaper. It could be the journal article. It could be the conference article. But my particular interest for today is journal article. But the journal article again could be different type, like the review articles, the case story, the commentary, or the research article. So I will emphasize particularly on the research article today. Any journal article, the main criteria is it must be peer reviewed. It must be reviewed by some intellectual non-expert or expert. Usually, editor, all journal has a editorial board, and one editor will be assigned when you will submit your paper, and editor will then assess the suitability of this paper. When editor will be happy, he will invite 2, 3, 4, 5 reviewers. Those reviewers will be very expert in that particular field, but the editor, not necessarily all the time will be the expert, but will be intelligent enough to understand your work. That's why I call them intelligent non-expert. Let's talk about the journal article. Journal article could be like full length. It could be short-com. It could be case story. The short-com is like 1 page or half page or 1.5 page, something like that. But the full length paper could be like 2 page to 4, 5, 6, or 10 page even. There are several parts of the article. I'm talking about full length journal article, research article. There should have a title. There should have keyword. There should be abstract. This title, keyword, and abstract, they should be accessible to all the people. Other than that, there should have introduction, but somebody call it background. Whatsoever, introduction and background, both are the same thing. There should have materials and method. There should have results and or discussion. Somebody just merged them together, results and discussion, but somebody discussed them separately. There should have acknowledgement, author contribution, some journal even asking for conflict of interest. There should have reference, tables, figures, and supplementary data. But the most important question, why do we publish? Is it necessary to publish? If yes, why? It is very important to share your data with other researchers. Let the other researcher to use your data. Sharing is very important in science. This is how science is growing. No science could grow alone. As you have heard all the day about next generation sequencing or deep sequencing, think about next generation sequencing analysis without a computer or without powerful computer or all this bioinformatics. Is that possible? Of course not. Think about the real-time machine, thermocycler, which is invented by some like smart chemical engineers or something like that. That means sharing your data and let other people reuse your data is very important. And other thing is, it's a kind of credit. It's a kind of contribution to the science. It's a kind of knowledge that you are creating. and disseminate this knowledge and share this knowledge with others. It is also important to justify your work. What you have done, is that worthy? Is it justified? So let the other people to evaluate your work. And this is how you will be evaluated your work. Important is to get recognition of your work. And this is how you can get the funding. If you publish, other people will know, other people will appreciate, and you will get the publication. Other important thing is promotion and award. For a researcher, it is very important to get promoted, like lecturer to assistant professor or associate professor or professor. Publication is the very important criteria or indicator to assess the researcher and to get the promotion. And for the student, of course, it is very important for getting their degree. So publication is the very, very important things if you want to be in academic and research. Let's talk about the different part of the publication. I'm not going to talk about the textbook things. I'm going to talk about my personal experience, what I have learned after working with all these different journals since decades. First, let's talk about the title and abstract. Title should be very smart. It should be short. Keep in mind, some journal even set the word limit for the title. So title should not be very boring and long. It should be attractive. It should be appealing and short and smart. What you should do, try to avoid any scientific term or any symbols in the title. Why? This will make it like something not familiar to the people, and people does not like that. However, it's not always possible to avoid the symbol. Like if you want to work with delta T cells or something like that, of course you have to use the symbols, but always try to keep it simple. Try to avoid very boring or unknown scientific terms. There is some tricks how you will choose the title. Some people are using topic as a title. Some people are going on the other way. The outcome is a title. I'll give you two example. Somebody used topic as a title, like identification of the genes for meat quality trait. So here their emphasis on the meat quality trait. But some people will make in the other way, like identification of the marker in IFNI6 gene, which is related to D+. So the outcome they are using as a title. Whatsoever, in either way, you should be very serious about the title. It should be informative and it should be effective. And then the author's name and their affiliation. How many authors could be there? There is no limit. It is painful that some journals are setting that, oh, there should be maximum 4 authors, but this is just rubbish. I don't like that. As many as people will contribute, they should be in the author list. And the affiliation. Some person could have more than one affiliation. And another important thing, I think most of you have this experience, the serial of the number of the name. Who should be the first author? Who should be the last author? Look, there are lots of things to be agree and not to be agree. So it's always depending on the discussion with your supervisor and with the team member, who should be the first author. To me, who has contributed most should be the first author. But I know the example where the professor is always being the first author. I don't know. Probably she has the explanation, but I don't have. And then the abstract. What is abstract? Abstracts actually cover everything. It covers your hypothesis. It is covering, discussing about the important results. It is talking about very briefly about the method. It is talking about summary. So abstract is like dense but concise, everything together. But as I told you, title and abstract and keyword will be available to everybody. So make the abstract very simple. Try to avoid difficult terms and terminology. Keep it simple. Write the abstract in simple language. One important thing, there is word limit or sentence limit for most of the journal. Like you will see mid-science, this says abstract must be not more than 250 words. So abstract should be very dense and always check the author instruction. That should have some limit and make it within that. The keyword. Keyword is very important because keyword will help the other researchers to find out your papers. So keyword could have some limit. Different journal will say different number, like most of the journal will say don't use more than 6 keyword. My suggestion will be find out the significant keyword which can help the other researchers to search your research and try to make it different than the title. Important message is title and abstract should be smart, concise, and in simple language because title and abstract is selling your paper. This is the main things that can sell your paper. Next, let's talk about introduction or background. Introduction is actually introduce your topic, what you are going to do. Tell the reader about your research, what you plan to do. Make a general statement about the problem, why you want to do this research, where's the knowledge gap, where you can contribute in science, highlight the importance of the problem, and then create some context and background in compare on the basis of the other research. That's what we call literature review. Talk about what other people done before, and on the basis of that, build up a very nice story, and then highlight the knowledge gap and talk about your research, how you can fit into that, how you can fill that gap, knowledge gap. Talk about why you want to do research and explain why your research is important. Talk about the importance of the problem and talk about the importance of your research. Stress the value and relevance of your research. Is your research relevant? Is it going to solve a problem? Is it going to contribute to the science? Why should a reader care about your research? Question by yourself and write the answer. This is how you can write a very nice introduction or background. As I told you, identify the knowledge gap and try to solve that. Make your research logical and record-based. At the end of the introduction, you will build a hypothesis. What you want to get, what you want to claim as a knowledge, and describe the important results that you may found or you are expecting to found. Outline the characteristics of your study. End of the day, introduction should be brief, clear, and concise. Here is the key take-home message. Introduction convinces the reviewer and editor and the reader. That's why it is the most important part of the article, to my sense. Don't talk about something that you are not going to do. Talk about something that you are going to do. Your introduction should have some link with your results and your discussion. Don't talk about that is going against you. Talk about something that you want to say at the end of your manuscript. That's why introduction is a part where you will convince the reader, you will convince the reviewer and editor. Don't say anything that you cannot prove in your results and discussion. That's why it is the most important things. Now talk about the results and methods. Materials and method very important. I guess this is the most or easiest part. Student interested to write it first. They find it easier. You can write the materials and method on the way you are working simultaneously. Important thing is you need ethical approval if you want to work with animals or something. Nowadays is very important issue because of animal welfare activists. So get the ethical approval. And in materials method, there are a couple of things you need to keep in mind. Talk about the subject. Are you going to work with cattle, sheep, goat, or what? Talk about that. When are you going to work? The time frame. Where are you doing this work? In which area, in which lab, or in which station? Talk about that. Very important things is the number of animal, the population number. It is most important in case of epidemiological study, but in other study also it is very important, as you have heard about from Asif, as well as from Amin, as well as from also Ulash. The grouping of the animal, the number of animal, how you group them. Let the reviewer to understand these things. If reviewer could not understand your experimental design, this will be catastrophic. I'm going to tell you, it will never go through. The selection of the animal. How many animal you have selected? Which tissue you have selected? Which sex, age, breed? Talk about all of these things. So another important things, resultant materials and method is talking about how you do the work, the procedure, the experimental design. An important thing is justify the experimental design. A good design is important, and more important is justification of the design, why you have done, what criteria you have set up to select the animal or for doing this work, what criteria you have selected to exclude some animal. So justify what you have done. And always refer, in case of materials method, like if you use normalization, sums in for normalization, always give the reference. If you use like DNA extraction following XYZ, always give the reference and talk about the modification, what the modification you have done. Talk about the procedure, talk about the instruments, reagents, kits, and chemicals, including the company from where you have purchased this one. This is very important. Explain how the results were analyzed, which software you have used, what analytical tools you have used, what statistical significance level you have set up. Talk about everything. And for all these materials and method, the tips is to write is start with the general information and then talk about the specific thing. Well, if I give you an example, it will be clear, like you have to talk about the animal first and then selecting the tissue and then DNA or RNA isolation and then sequence. If you talk about the sequence first and talk about the animal last, that does not make sense. So there should have a sequence. And always mess up with the order of the methods with the order of the results. The order you will do the work, in the same fashion you will write the results. That's why when you are doing lots of work, and that's what nowadays people are doing, always do the subsection. Always include the citations of the procedure if you follow somebody else. And as I told you earlier, also mention the modification if you do any modification. Statistical analysis is very important, and important tips to you. Avoid discussing pros and cons of certain method. Don't blame any method, just do your work. Don't talk about the good or bad about that method. And important things is the method should be repeatable. If some other people read your method, can they repeat the same work? That's the purpose of materials and method. But what I find that it is the easy target to reduce. When supervisor is asking, oh, your papers is getting long, so make it smaller or reduce the text, students are first going to cut the materials and method. Whatsoever, method should be concise and yet sufficiently thorough. Important thing is materials and method attract the researcher. Who is going to do similar type of research, they always look to your materials and method. As I told you, this is the easiest part. The student always think about and they wrote it first. Now let's talk about the results. Results is the outcome that you have found. Result could be the effect or the indicators of the solution. Important things is only the findings of your study should be here. Don't talk about other people's findings. Absolutely, your findings should be in the results section. And other important things, present data in tabular form or chart or graph or some sort of visual form. That's what people like. That's what readers write. Visual form could make it easier. So present the data in tabular or visual form. And when you are writing the results, correspond the results to the key questions. Why you are doing these research? Always keep that question in mind and highlights those type of results. Always highlights the most significant result, the statistically significant results. You don't need to discuss all the results you have found. Other thing is, that's what I always find, that what is in the tables or in the picture, in the visual form, that is not always messing up with the text. So always double check. Like some people are talking, this gene is high expressed in this animal, but if you go through the table or figure, it's actually on the other way. So don't underestimate your results. Don't underestimate your writing. Always double check the text and the tables or graph or whatsoever. Another thing is always ensure that your results are represented in the best order. When I talk about the best order, as I told you, the materials and methods should have some connection with the results, and results should have some connection with the discussion. Always talk in the same fashion, in the same order. So there will have some easy reading and some connections. In results, don't make any recommendation. That's not the part should be discussed here in the results. Refer to the appropriate table and figures. And nowadays, since most of the journals are online, you can add up supplementary data, which you think is very important, but very big or not most important to highlight in the paper, but you can submit as supplementary data. Other thing is prepare the tables and figures in most informative way and in dense way. Try to merge 2-3 figures together if they are conveying the similar type of message. This is very important and easy for the reader. Don't explain each and every data. That will be the repetition. People can go through the figure or table, so you don't need to explain each and every data. Explain the most important, significant, statistically significant data, and explain those results, which is going to answer your question, your hypothesis. Now the discussion. I think discussion, most of the people find it the hardest part, but I don't think so. Discussion is the things where you are explaining results. how it is fitting with your hypothesis. You are comparing your results with other people's results and then draw a conclusion. But in most of the cases, what we see that discussion is very repetitive, it's very loose and very large. That's not necessary. Make it dense and make it not too large, not too small, and always be concise. Make your points clearly. Avoid using a repetitive word. And as I told you, there should have some order in discussion too, like in materials and method. There should have a logical stream of thought. Let the reader read it easily and justify your outcome. That's what the discussion is. Discussion is all about justifying your outcome in light of your hypothesis. Establish the fact that you want to establish. Refer to the specific work or the work that has been done that is in line with you. If needed, use the subheading, because when you are doing lots of work, you need to subheading the materials method as well as discussion. So some sort of connection should have between the materials and method and the discussion. Explore the underlying meaning of your research. What your data is mean, what your data is representing. Talk about that. Which one is high or low? People can easily understand, but talk about why it is high or low and how it is fitting with your purpose. That is most important in discussion. Always think about the outcome. Is it logical? If you find the data a gene expression which is high in good animal, but literature says that it is not a good gene, don't talk about that. Always talk about the results that make sense and which is supporting your hypothesis. Otherwise, it will be suicidal. In the discussion, highlights the importance of your study, how your study is contributing in science, how your study is fixing the problem, how your study is filling the gap in knowledge. To do that, compare your results with other, and you will find that it is always not supporting like what another people got, you may find in another type of results. So if it is supporting, that's good. If it is not supporting, give an explanation. Like X found Y gene is high in good animal, but I found in not high expression. That is because of maybe we have used different breed of animal, or maybe we are using in different season. So always try to find some explanation why your result is not messing up with other. If it is messing, if it is in the same line, it is perfect. If not, always try to find out some explanation. Always try to find what makes the result different in your case than the other case. Why not in line with others? Always explain that. Analysis, and other thing is always talk about the unexpected findings. Explain the limitation and weakness of your research. That's what I always do believe, that don't let the reviewer to find the problem in your paper, the limitation in your paper, the unexpected findings in your research. Better you find the limitation and weakness and unexpected findings and then explain in the discussion. That's how the reviewer will be happy that he is not skipping, he is confronting. That is very important. What I want to call the self-critical. Criticize yourself, criticize your results by yourself, and always avoid apologetic type of things. Just be honest and criticize yourself and try to explain the results in your best way. Discussion is a good place where you can explain or your opinion where you can say what you want to say, where you can speculate the results. So you can forecast here. You can always suggest the future study, but don't suggest something that you can do within your scope. Then reviewer will ask why you are not doing. So suggest something that is important, but you cannot do at the moment in this study. Other thing is conclusion. In some cases, people are writing conclusion and summary separately. In some cases, this is the last paragraph of the discussion. In either way, it's fine, but there should have a conclusion. Conclusion is the take-home message, what you want to say to the people. It should be in very plain language, like abstract. It should be very clear. It should be very dense and concrete. And summary actually include everything. It is the sum up of everything. You have to remind the author about the hypothesis you are working on. You have to talk about the problem you are looking for to solve, and summarize the main point, the main results you have found. State the most significant results that is going to solve this problem, that is going to fill up this knowledge gap. And then your concluding thought, what you thought, is it worthy? Can this polymorphism could be used as a marker or not? This type of things. Always write a concluding sentence that represent your thought about your paper. Don't repeat the discussion and results that people have already read. So in summary, don't repeat anything that you have already talked in results and discussion. You can offer new approaches, but as I told you, don't say something that you can do within your scope. Like you can say, more study should be done in another population. Indicate the opportunity for future research. What the next student or what the other student or what the other researchers can do further on the basis of your study. Talk about that. As I told you, don't offer recommendation which could have been easily done by you. Then reviewer will ask you why you are not doing this by yourself. Remind your reader the strength and the impact of your argument in the conclusion. As I told you earlier, summary is the take-home message. So it should be very dense and it should have your own thought. Not all the case. In case of review article, the conclusion will be different. But in case of research article, conclusion should draw on basis of your work, not other people's work. Now let's talk about the references. I always believe that credit should go to the original article, but students always try to write the cross-reference. They don't want to go to the original article. How many references should be in an article? It depends on the type of article. Like in research article, 50, 60, 70 could be okay. But in review article, it could be like 100, 200, does not matter because they're talking about all the previous work.
Hello, Doctor. Sorry, we have only less than 5 minutes left, Doctor.
Oh, okay. I'll maybe need like 10 minutes. And then for the style, always see the journal. And usually there should have like author name, year of the publication, title, journal title, volume, issue number, page number. And my suggestion is always try to use software like EndNote, Reference Manager or something like that, which will make your life easier. There are other parts of the article could be like funding. It could be like grant, scholarship, or anything, author contribution, and table, figure, supplementary data, all should be in the articles, of course. And another is most important things I think you are just waiting for is the rebuttal, the reviewer response letter. Don't think that reviewer are your enemy. Don't think that reviewers are the villain. They are there to help you. Reviewer and editor, they are doing this free service. They're not getting any money for doing this review. And so believe me, take it positively. Don't take it negatively or personally. Reviewers are always there to help you. Reviewers are trying to find out the gap that you should do to improve your paper. We have lots of experience that we submit, it gets rejected, but reviewer give very nice comments. On basis of that, we finish this work and then we publish in better journal. So take it always positively. What should be in the response letter? In the response letter, always appreciate the reviewer and editor for their comments, for their time, because they are doing this free service to you. Respond each and every point. Don't escape any point. There is no hide and seek. Be polite and respectful. Don't say, oh, this reviewer does not know. Why you don't know this one? No, always defend. Don't try to tease the reviewer. So always write the response letter in passive way, in polite way. Accept the blame and apologize for that. There is no wrong with that. Say, yes, sorry for this spelling mistake. Yes, sorry for these things. Always apologize. Accept the blame. Confront it. Make the response self-contained or self-explanatory. Don't say, please see the response in another paper. That's not the reviewer work. That is your work. You have to mention what you want to say here. So it should be self-explanatory. It should be easier for the reviewer. And there should have some linkage between the reviewer response and in your paper. If you reviewer could just escape something or overlook something. So always say, please find in my article at page number this. So always navigate, always help the reviewer to find the information he's looking for easily. Wherever possible, answer directly. And when possible, do what the reviewer asks. If reviewer asks you to do additional work to quantify one more gene, if possible, within your budget limit, do it. It will convince the reviewer. Be clear about the sense. Nowadays, most of the journal, like any open access journal, they will ask you to use the function review so that what sense you are doing, they could track and then they could see. I mean the track sense, review with track sense. In the response letter, you can add up reference. There is no wrong with that. Use the supporting evidences, which does not necessarily need to go in the main paper. Like if reviewer is upset about or confused about the primer, whether it is working or not, just run in a gel and put the gel picture in the response letter. You don't need to put that in the main paper, but you can put it here. However, it should be comprehensive and straightforward. So where to publish, how you will select the journal. That's the last part. I'm going to finish in a few minutes. Selection of journal is always a tricky thing, but my suggestion is write the paper first, and then you will know who is your target reader. It could be specialized, it could be interdisciplinary, like the meat science. Only if you're working with meat, you can publish in meat science. But in like animal science journal, meat science, production, anything you can publish there. So write your paper, think who is your target audience, and then publish in that type of paper. Always be careful about the credentials of this journal, because nowadays there are lots of pseudo journal. I'm going to talk about 1 minute later. Think about the quality of their website. Think about the quality of the paper. Always check the website. Are they publishing any paper which seems like you? Are they going to publish paper of your types? Is there any relevance of your paper with that content? And try to publish in the appropriate field. Check the journal. their scope and their policy should be clearly mentioned in the author instruction. If your research is not fitting in the scope of the journal, definitely they will not publish it. They will straightway send you back after submission. So like in animal science journal, you cannot publish like something done in human. So always look the scope of that journal and read the author instruction very carefully. Look their peer review process. Look their importance. Look how much time they are taking for reviewing. Look the acceptance and rejection rate. All this will help you to select a journal. And important things is always add up a cover letter when you will submit the first time or when you will submit the review response, and submit your research to one journal at a time. Otherwise, this will be just very bad thing. Follow the author instruction carefully. Prepare the manuscript according to the author instruction. Check whether it is open access or traditional journal. Time, how much time do they need? Think about that. Sometimes you could be in rush, and cost is very important. Some journal is getting really expensive nowadays. So you should also think about the cost of the publication. Other thing is citation. This is the most famous thing, right? Let's talk about some citation. Always look for the reputation of the journal. Don't go for the predatory journal. Nowadays, you will find 100 and 1,000 journal in the online. They are just predatory journal. They are gaining their financial profit from the author, but they are not maintaining their standard at all. How you will find the predatory journal? If you Google, you can easily find out. You will see that they are frequently sending you email. You will see that they're promising that your paper will be getting publication within few days. They will sometimes negotiate the fees. So you can easily find out which one is predatory journal. Be careful from predatory journal. And impact factor. I'm not very much like this impact factor things, because impact factor represent the relative importance of a journal. It's not absolute, it's relative. And it's very much field specific. And it is talk about the average article, not about your article. It is talking about the particular time period, how the journals or the average of the articles of that journal is doing. That's why you will see one journal impact factor is going high in this year, next year is going low. I know that you all guys know how the impact factor is calculated. There is an example here. You can easily find. You know, the main purpose was to invent this impact factor was to create an indicator for the librarian. Nowadays, all people are using impact factor as a gold standard, but it is not a gold standard, I'm going to tell you. It is a very relative thing. It's very much relative to the size of the particular science community. Like in human, there are huge number of people working. In animal science, only a small group of people are working. So it is all about the citation, and citation is all about the size of the community, science community. So don't run after the impact factor. Think about the field specific, good, reputed journal. Because impact factor is not going to tell anything about your article. If you get in publication in a good journal, but if your article is not getting citation, it does not make any sense. So how do you publish? That's the last slide. My suggestion is read the author instruction carefully. Read and write. Do it daily. Make it a daily practice. Don't cut, copy, paste. That's the most easiest things, right? But don't do that. Read and write daily. On the other hand, journal space is not unlimited. Although the online journal will give you more space, but don't make it too long. Read your draft carefully to catch up any language error, grammar error, spelling mistake, or typo, or anything that is not fitting with the papers. So read it carefully. Here is my suggestion. Read, and left behind. Forget about these papers for a few days or few weeks, and then get back to the paper again and read, rewrite. You will find a lot of problem by yourself, and then rewrite it, and then this is a circle. Read again, left it behind, and read and rewrite. Get the feedback from your supervisor, from your advisor, from your colleagues. This is how we get the third eyes, right? When you are reading your paper day by day, every day, your brain and eyes are actually sitting with you. You will skim through, you will not read all the letters, all the words, so you will not find the problem. So get other people, I said the third eyes, to jump in and find the problem. Wish you all the best. Oh, sorry.
Thank you, Doctor, for a very interesting talk on how on the tips how to publish in a good journal, and also how to address the issue raised up by the reviewer. All right, I think we're far way behind the schedule. I just want I go directly to the Q&A sessions. I have received several questions already for both speakers. Prof. Mehmet is still here, right? Okay. Hello, Prof. Can you hear? Can you get my voice?
Ulash.
All right. Okay. Okay. I think I will go first to the first speaker for the questions. Let me check. There are two questions compiled. I think I select only two questions. The first one is for Prof. Ulash. According to my knowledge, from Sigit Prastowo of UNS Solo, there are two methods of QRT-PCR, namely Delta-CT and quantifications method. Among those, which one is better or more acceptable in publications among the Delta-CT and quantification method, or any other considerations in the QRT-PCR reactions? Okay, maybe you can answer
yes.
first.
So could you please repeat the question?
Okay, among two QRT-PCR method, the Delta-Q, the Delta-CT, and also the quantifications, which one is actually more preferable for publications? Or do you have the option?
Yeah, so I got it. Thank you. So actually, so it's upon your, how can I say, so upon your aim. So there is no means that there is no preferable one. So this is one of them is not more preferred by journals. So it doesn't mean that one is preferred more than the other one. It depends on your aim actually. But so the standard curve method is mostly used to, how can I say, so mostly used to identify the actually copy number. So the messenger RNA or so cDNA or DNA copy number. This is mostly used in my opinion. So in my opinion. So both of them is okay, but in my opinion, the copy number is more so required for example, pathogen detection. Means that bacterial load, or for example, so in the plant science especially, so the viral load, bacterial load, fungal load, or in the animal, for example, so disease detection. So for example, now I'm working with one Q fever. So for example, the Coxiella C. burnetii bacteria load, we applied, for example, the how can I say, the standard curve method, because we need the absolute copy number of the bacteria in the tissues. Yes. But if you want to just to quantify or validate the gene expression, the delta-CT method is, so it's enough actually. So, and journals are also accepting both delta-CT method, but if you would like to show the, so really the copy number of the molecule, gene, messenger RNA, bacteria, whatever, yeah, so you need to make the standard curve method. It depends on your, how can I say, on your approach. Yeah. So what you would like to show. Both of them is accepted by journals.
Okay. Right.
Hey, can I jump in? Can I add up?
Sure.
So it's depend on how much money and time do you have.
Yeah, that's also.
So if you have money and time, you can go for the standard curve. But if you want to do it quickly, just do the delta.
Yeah, but so one more thing, just so if you want to show for example pathogen load, so delta CT is not enough. Yeah, so this is must.
Yeah, that's
so
right. That's right. It can only compare.
Yeah, exactly.
The second question is from Prof. Irma. In regard to the RNA work for bacteria, do you have any experience in RNA-seq for the bacteria, whereby the aim is to identify specific bacteria that can contribute to the changes of the quality products in the food products, let's say flavor changes or something? Is there any way we utilize the RNA-seq approach or RNA-works approach for such kind of purpose?
Is that question for Aminul or Ulash?
For Prof. Ulash first, Doctor.
Okay. So yeah, I personally don't have experience. So I never work with the microbial microbial DNA or RNA. I mean that whole genome sequencing. Actually, I never done that one because I'm not veterinarian or I'm not, how can I say, microbiologist actually. Yeah. But it is possible. I know that from the literature, it is highly possible. Yeah. So you can how can I say, you can isolate the DNA or RNA from virus, bacteria, or fungus, for example, and then you can, how can I say, make the whole genome sequencing actually. Yeah. So this is possible. Yes.
Okay. Thank you.
Yes. Sorry, this is possible, and it is using a lot. Lots of people are working with this metagenomics type of things, sequence the whole bacterial product, whole population, and then up with the existing sequence, and then you will get up with nice curve or something like that. What can say the population, which type of bacteria is there? Yeah, lots of people are working with that.
All right, thank you, Doctor, for the additions of the explanations. Okay, now the question is for you, Doctor, for the second speaker, for Dr. Jachim. All right, the first question is, somehow sometimes we face the reviewer who has a conflict of interest with our paper. Okay, how do you handle such kind of issue? Even though we believe that we have wrote the manuscript well, but due to the conflict of interest, then the reviewer rejected the manuscripts.
Oh, that's I will not agree with you. If the reviewer is very fair,
okay,
accept the invitation. First of all, I'm going to tell you, when the journal is sending any paper to me, they will first send us details about the author. Do you need this author? Are you working with this author? Do you have any personal contact or any financial relation with this author? If you say yes, they will not send it to you for the review. So when any reviewer is first inviting, of course, they will send this type of information. If any reviewer, if he is fair, you should not accept the invitation. So not with the reviewer.
But
if some reviewer is not fair, he is lying and he is accepting the invitation and reviewing your paper, and in any way you came to know that I know this guy, he is our enemy, he is working in other university or other lab, and they are always fighting with us, and he is the reviewer. Talk with the editor, he should kick out him.
Okay. All right, thank you, Doctor. The second question for you is in regard the way we defend. How do we defend ourselves when we have such constraint to repeat the experiment that requested by the reviewer, but due to the constraint of the instrumentations, due to the constraint of budget and so on, we are unable to co-opt that comments. Yeah. What is the best way to address?
Yeah, that's what I try to tell, that when they are asking, reviewer is asking any questions or any additional work, if it is possible, do it. He will straight away accept your paper. If not possible, just find an excuse. Write very polite way. Thank you, reviewer. If we could do that, we do believe that that would improve our paper. However, we have the budget limitation. However, we have time limitation. The tissue is used up. So make some excuse. Don't be arrogant that, no, I don't want to do that. No, I don't want to repeat it. Try to find some excuse, try to find some papers that have done similar work to you. Use that as a reference in the response letter, not necessarily in the main paper, but give some evidence that what we have done, we have used up like 3 animal. There are a lot of other people have used 3 animal. So what's the wrong? If they says GovDAIS is wrong, you says, no, there are lots of people are using GovDAIS, so we have used. Don't be arrogant, be polite, try to convince him.
All right, okay. Thank you very much for both speaker, Professor and Dr. Jasim for very good presentation for second sessions. Unfortunately, we have very limited time and we are already behind schedule actually, but I think because of the interesting topic, the participants still enthusiastic to follow. So I regretfully would like to close the second sessions, and before I end up also all the sessions, I would like to share the certificate appreciation for the speakers in the second sessions, and I would love if, okay, a board of speakers still hold with your video, so we can take the documentations. Allow me to share the certificate of appreciation for both of you. All right, okay. I hope you can see my slide, right? Okay, the first certificate goes to Professor Dr. Mehmet Ulas Sinar for being a speaker in today's webinar. Thank you very much, Prof. I know it's early morning in Turkey, and I really appreciate for your time to be with.
Thank you very much. I thank you. So I enjoyed a lot actually. So it's my pleasure to meet you actually.
Thank you, Prof. Thank you. Really appreciate it. And then also, of course, for Dr. Associate Professor Muhammad Jasimuddin, DFM MS PhD, for being with us today. I don't know what time is it in Bangladesh now?
Oh no, I'm not in Bangladesh. I'm in
Australia. Yeah,
another +4. It's +4 p.m.
Okay, okay. Thank you again for being with us, and I hope that we can have another chance to meet again. And in other event. All right. I think that's all from me. It's now 1 p.m. already, 1:17 p.m. already, lunchtime already here. And before I close this this morning webinar sessions, I'm still sharing my slide. Okay. I'd like to stop here. All right. Okay. So I think I regretfully should also stop the webinar program here. From my side, I don't think I need to conclude anything. Basically, if you want to do the RNA-6 work and whatever, so just follow all the advice from all speakers, and also whenever you publish your data in any journal, to get your data published well in reputable journal, please also follow the advice from the last speaker. Yeah? So again, my highest appreciation to all speakers and participants. I wish that today's webinar is fruitful for everyone and also for the speakers. And of course, certainly it's very fruitful for the organizer. On behalf of the committee and also myself, I would like to extend my sincere apology for any inconvenience during the event. And before I close, I would like to ask committee, is there any announcement? Pak Arman, ada slide yang mau di-share? There are some announcement and promotion that I would like to share by the committee. Okay. Where is the PowerPoint? PowerPoint-nya, Mas? Okay. while waiting for okay, all right, okay. So we are now in the series 5, the 5th series of the webinar for today's webinar, and we will still have for the 2 coming webinar soon on July 13th and July 28th. Both will be in Bahasa Indonesia, unfortunately. On July 13, we have deteksi kulit hewan asli secara cepat dan akurat with Professor Dr. Ono Suparno dengan Dr. Maria Ulfa. And then lastly, on July 28, we will have perspektif kesejahteraan ternak di masa adaptasi kebiasaan baru. 3 speakers will be in the series 7 of July 28. Prof. Dr. Zulkifli, Dr. Hewan Wibik Bagja, and also Professor Dr. Iman Rahayu. And then next slide. Okay, please, the participant, please join also the program. So this is the flyer for the next coming webinar on July 13. You can register on the following link. Yeah. Next. And also, the department also would like to call for admissions for the postgraduate program under Animal Production Science for the academic program of 2020-2021, which starting from 13 May actually until 28 July. We'll close on 28 July. And then the selections announcements will be on 14 August on 2020. You can see the link here. You can register either as a regular students or by research students. Okay, next. And then also call for paper. The department or the Faculty of Animal Science run an international journal called a Tropical Animal Science Journal. It is Q2 journal, and it's open for anyone around the world to submit their papers for this journal. Okay, next. And then the journal under the department, it's Jurnal Ilmu Produksi Teknologi Hasil Peternakan. Is indexing in several indexer, including SINTA. This I think is suitable for those from researcher from Indonesia. Next. And also acknowledgement for all the team of department webinar, as advisor, Professor Dr. Ima, and then the lead is Dr. Asad Gunawan, and the rest are the members, Dr. Tuti, Dr. Zakia, myself, and Dr. Yuni, Mr. Ferika Armansyah, Jimas, and also Dr. Astari. I think that's all for the announcement. Is it? Anything else? Sudah ya? Next. Sudah. All right, okay.
May I have one question?
Sure, Prof.
So are you accepting papers for tropical animal science from the subtropics?
Yes, basically it should be no problem. I think Dr. Asaf should be happy with that. No problem. All right, okay, thank you. All right, okay. I think, okay, then by Alhamdulillahirabbil alamin, then I would like to close today's webinar and hope to see you again in the next coming event. But before you close, please, all the participants, can you turn on your video for a few minutes, because I think the committee will take what you call it, print screen all the picture from all the participants attended here today for the documentations. Can you turn on your video, please? Okay. While waiting the committee to take the picture, for myself, I think again apologize for any inconvenience as a moderator for today's webinar. Thank you very much for being with us. Okay, Mas Arman, silakan Mas dipandu saja, atau Pak Asep. Oke, masih ya. Okay, so the committee is now recording the picture. Please hold your smile for a few minutes.
Okay, thank you. Udah.
Oke, Mas.
Oke, udah Pak, udah.
Udah semua?
Sudah Pak, sudah.
Alright, okay, the recording is done. Okay, thank you for your smile And again, thank you for the participation. Thank you very much and good day for everyone. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam. So thank you all participant, Prof Ulas,
Thank you, Prof.
Dr. Jasim,
Thank you, Dr. Jasim.
Dr. Amin for sharing your knowledge and all participants
Thank
from
you, Professor
different
Menmen.
university.
Thank you, Mr. Agus. And thank you for Dr. All. Salam cinta dari ujung kandang, Viva Veteriner Mania.
Terima kasih.
Hello, Sam.
Terima kasih.
From Australia. Hello, Sam. Thank you.
Ya, thank you.
Thank you,
Ulas,
Rafida.
Dr. Jasim, Dr. Amin, Bapak Ibu semua, terima kasih semuanya.
Terima kasih.
Hadir di webinar kami.
Terima kasih panitia, Pak Asep.
Makasih Pak Nafiu, Pak Dr. Nafiu. Ya.
Terima kasih, Pak.
Sama-sama, makasih Pak. Ibu, makasih. Bu Heni, makasih Bu Heni. Pak Rusdin, makasih. Ibu Yolanda, Sam, thank you.
Terima kasih, Pak.
Ya, sama-sama Ibu. Sam, thank you.
Makasih semuanya ya.
Ya,
Sama-sama, Ibu.
Purpon, makasih Purpon.
Terima kasih, Prof. Suyadi.
Terima kasih Pak Asep.
Sama-sama.
Terima kasih, Mas Purpon.
Terima kasih Bu Anggreni, Prof. Suyadi, Bu Nova, Pak Dudi. Terima kasih Pak Dr. Dudi dari Unpad.
Izin live ya.
Ya, makasih
Ada,
Ibu
Bu.
Yolanda.
Makasih ya, Bu.
Ya, terima kasih ya Bu. Sampai ketemu lagi Bu.
Ya.
Makasih. Siapa lagi ini? Pak Bayhaki. Terima kasih Pak Bayhaki.
Makasih Pak Asep.
Ya, makasih Pak
Makasih,
Rusdin
Pak
ya.
Rusdin.
Ya, sama-sama. Terima kasih. Ya.
Gimana Bu?
Pamit,
Ini
Pak.
kita
Assalamualaikum.
Waalaikumsalam Pak. Ini kita live langsung Bu. Kita end.
Ya, kita end.